big hello to all my PU2 champions. How are you all? I hope the Josh is really high, and I welcome you all to the marathon series. So today we are going to deal with all the one marker question. So one marker as in we're talking about MCQs. Now MCQs are considered to be very lucky. Okay, why? Because we do the guesswork and depending on the luck we get the mark. But this time you would be lucky because you already know the answers, because we would be discussing a lot of questions from all the chapters in your NCERT. There's 13 chapters in your NCERT. Now disclaimer, this video might be long, but it will be really useful, and focus on the next word, that is useful, my dear children. Okay, do not focus on long. If there is something that is useful, that is going to give you marks, then why not just watch it up? And obviously we are putting our 100% efforts. That is why we are standing and recording this particular video. Now some of you might ask, why not live? Because in live there could be distractions from your end as well, okay, or probably I might be explaining a lot and I don't want that. Okay, so I want to save as much time as I can because you guys are preparing for your exam and I know that. Okay, so the schedule is here, my dear children. On Wednesday, Feb 19, model paper 1 was released. Now Thursday, Feb 20, this particular video, that is biology all 1 marker. Now Saturday, you have 3 things coming up, model paper 2, biology all 2 plus 3 markers, and also biology PU2 all diagrams. So I saw a comment down, ma'am, please can you give me biology all diagrams? So here is it. Okay, here it is. So please don't worry, my dear children, all your queries which you are putting down in the comment section, one or the other day it will be sorted and it will be done before your exam. So you just have to put down in the comment section what you need from us, and all the doubts also. The doubts that you're going to post today, we would be taking this on priority and solving it before the day of your exam. So whatever doubts you have, we would be taking the screenshot and putting it down and solving it in live session before the day of your exam, that is biology exam. And now biology of 5 marker will be on Monday, and the passing package will be done on Wednesday, that is February 26. So I hope you are updated about the other teacher schedule. I would be giving down the lane, okay, while we are discussing. We are done with one chapter, then I will be talking about their schedule as well. Chalo, I don't want to waste your time. So first chapter is sexual reproduction and flowering plants, my dear children. I have included 10 questions here. Okay, now first question is, dioecious plants are, what do you mean by dioecious plants? Dioecious plants as in the flower, my dear children. One particular plant would be female plant because it would be containing flowers that have the female reproductive structure. Other plant would be having the male reproductive structures, which is why I would call this a male plant. So over here, dioecious as in 2 plants are there and the sexuality is divided among 2. So over here, will you be seeing gytonogamy, autogamy or xenogamy? So listen, xenogamy basically means whenever pollination happens between 2 plants, that is xenogamy, and that is the only option that suits over here. So your answer becomes xenogamy. So you have to look for xenogamy. What do you mean by gytonogamy? I'll be talking about that also. Gytonogamy basically means if you have 2 flowers on the same plant, from here to here things are getting transferred, pollination is happening, that's when you call it gytonogamy. But here also you have female flower, here also you have female flower. Do you think gytonogamy can happen? No. So that option is wrong. Now autogamy means basically from the single flower, from the male parts to the female parts, things have to be transferred. But here there is only the female part. Again, this cannot be happening. So in dioecious plants, I will say xenogamy is the only option that they have. Why is it not working? Chalo, it's okay. So this is the answer, okay? Next question. Endosperm persists in the mature seedling. Now, my dear children, endosperm is something that is made, okay? Endosperm is made to provide nourishment to who? To the zygote. Because the zygote would have to transform into what? Embryo. But while this happens, the embryo says that I am going to use up all the endosperm for my own nutrient storage. So what it does is that it stores nutrients in the form of something else that is known as cotyledon. So in some type of embryo, they are going to have 2 cotyledons, so they are going to utilize the whole endosperm. In some other plants, they are going to take some endosperm and make 1 cotyledon out of it, but they would be also leaving some endosperm back. So there will be some endosperm that is left. Apart from that, there will be 1 cotyledon also. You call these plants as monocots and you call these plants as dicots, where there are 2 cotyledons. Now how do you signify monocots and dicots? Basically, you can understand if you can break something into two, they are dicots. They are the cotyledons. The two things that you're going to break it down into is the cotyledon. For example, pea you can break it down into two. You have chickpea also, you can break it down into two. Beans, you can break it down into two. Groundnut, you can break it down into two. All of them are dicots, my dear children. So in the monocots only, please remember, in the monocots only, you would be having endosperm that is left behind. Okay, so over here, groundnut is there, so this is not an option. Pea is there, this is not an option. Beans is there, this is not an option. What do you have? Castor and coconut, both of them cannot be broken down into two. Basically, they are monocots. So you might be thinking, ma'am, coconut, if I throw it, it can be broken down into two. Not like that. Okay, not like that. Basically, obviously, you can break anything into two in that sense by throwing. Okay, so this becomes your answer, my dear children. And you call these type of seeds, no? You call these type of seeds as albuminous seeds, where the endosperm is still left. Okay. So with this, you get two information. Now, next question. Statement 1 says, in Viola, both autogamy and geitonogamy is prevented. What do you mean by Viola? So there are three plants, Viola, Commelina and Oxalis. Okay. And these three plants have two type of flowers. One flower looks like this, where it is totally closed, my dear children, but they have the female and the male reproductive system right over here. So over here, anyways, the pollen grains can land over here and cause pollination to happen. Right. Now, this type of pollination from within the same flower, it is known as autogamy. So can I say autogamy is definitely happening? Yes. And these type of flowers are known as clistogamous flowers. What do you call it? Clistogamous flowers. Now, talking about the next thing, they also have one more type of flower, which is open flower, having both the male and the female reproductive things. Okay. So here also I am going to make flower like that. So now from here to here, autogamy can happen. Again, from here to here, geitonogamy can happen. I told you, within the same plant but within the different flowers, when things happen, you call it geitonogamy. So autogamy as well as geitonogamy is possible over here in the case of Viola. So in Viola, both autogamy and geitonogamy is prevented. Is that true, my dear children? It is not true. It's not prevented. It happens. It happens, okay? It is not prevented, it happens. So, geitonogamy is similar to autogamy since pollen grains come from the same plant. I will say genetically, yes. Genetically, yes, because obviously you are taking the same plant's example. So, genetically it will be same like autogamy. But actually it is a form of cross-pollination. Actually, this geitonogamy is a form of cross-pollination, but genetically if we are talking, it is, yes, similar to autogamy. So, I will say statement 1 is false, but statement 2 is correct. Okay, so in Viola, both autogamy and geitonogamy is prevented. That is wrong. I think I marked the wrong statement, but I'll tell you the correct answer. So this is incorrect and the other one is correct. So statement 1 is incorrect and statement 2 is correct. This is the answer. Let me see what I have marked. Oh, I had marked that only. Okay, I read the question properly. I thought I didn't. So yes, my dear children, you have to read the question properly. So I was in a confusion that I might not have read the question properly. Cool, done. Let's go. Now select the incorrect statement with respect to vegetative cell of pollen grain. So you know pollen grain has a large vegetative cell. Inside the vegetative cell cytoplasm, there lies a small cell, that is the generative cell. This generative cell is the one that will give rise to male gametes, and this vegetative cell which is there, my dear children, it provides nourishment to the generative cell. Now you are asked about the incorrect statement with respect to vegetative cell, this cell. So, it is bigger than generative cell. Yes, it is correct. Has abundant food reserve. Yes, obviously. Has a large irregularly shaped nucleus. Is it correct? Yes, it is absolutely correct. Floats in the cytoplasm of the generative cell. No. It floats in the cytoplasm of the generative cell. No, generative cell floats in the cytoplasm of the vegetative cell. So, this becomes the incorrect statement that you are looking forward for. Okay. Fine. So yes. Now, which one of the following statement is not true? Not true. Underline. What you are searching for? You are searching for not true statements. In MCQ, I always say underline the key terms which you are looking forward for. So, pollen grains of many species cause severe allergies. Yes, obviously, a lot of people have asthma, bronchitis and things like that. Stored pollen in liquid nitrogen can be used for crop breeding programs. Yes, because in liquid nitrogen, they are quite safe. So, they can be used for crop breeding programs. Now, tapetum helps in dehiscence of anther. Is that true, my dear children? Tapetum is a layer, okay, tapetum is a layer in your microsporangium structure, in the anther structure, no. So, over there, you know, This is sporogenous tissue which is going to give rise to pollen grains. Now to nourish this, you have a layer that is known as tapetum. Now tapetum has nothing to do with the dehiscence of anther. Outside the tapetum there is middle layer, there is endothecium, there is epidermis. These three layers which are there, they help in the dehiscence of anther. What do you mean by dehiscence? Basically the anther breaks open and will be releasing all the pollen grains up over here. So tapetum is there for nourishment of this pollen grains, whereas these three layers which are there, they are there for dehiscence of anther. So I'll say this is a wrong statement. Now axine of pollen grain is made up of sporopollenin. Do you agree? Yes. Now when we were talking about pollen grains right over here, my dear children, okay? Yes. So pollen grain, it has its own plasma membrane. Outside that there is an intine, okay, the inner layer, and outside that there is an exine which is discontinuous. Okay, this exine is made up of a tough substance which is known as sporopollenin. So there was a question which I removed because I thought I will explain it over here. Okay, so sporopollenin is that structure which is there in the exine and it is resistant to alkali, acid, lots of things. And where you do not find the sporopollenin, there you find germ pores in the pollen grains. Cool? Chalo. So this is the answer. Going ahead with the sixth question. From the statements given below, choose the option that is true for typical female gametophyte of a flowering plant. So what is there about the female gametophyte, my dear children? Female gametophyte has three things to be noticed about. See over here. So they have micropylar end, okay, and they have chelazal end. In the chelazal end lies three very boring cells, okay, which are known as antipodal cells. They are antipodal cells. So they are anti-enjoyment also, they are just there, okay. Now over here in the micropylar end, you have the egg cell and also you have their best friends who are synergids, my dear children, okay. Now these three cells together, I will call it egg apparatus. I would call this as egg apparatus. Now there lies, now all of them have their own nucleus, okay. Now there are two nucleus which is just lying. I am talking about nucleus. Just nucleus, two nucleus are there just lying over there. And this is a part of a big cell which is known as central cell. So these two nuclei which you see over here, they are known as two polar nuclei. Okay, and this cell is what? Central cell. So if I count the number of cells, 1, 2, 3, 4, 5, 6 and this whole thing, 7. I have 7 cells. But how many nuclei do I have? 1, 2, 3, 4, 5, 6, 7, 8 nuclei I have. So basically it is 8 nucleate and 7 cell at maturity. This is absolutely correct. And you have to choose what? Choose the option that is true. Okay, so this is true. Now it is free nuclear during development. Is it free nuclear as in the nucleus is not covered by the cell wall and everything? Yes. So during development the nucleus keeps on dividing. Slowly the cell wall formation happens, my dear children. So this is also entirely correct. Now it is situated inside the integument but outside the nucleus. Is that so? No. So suppose this is the embryo sac. Now embryo sac is within the ovule, my dear children, within the ovule. Now ovule will be having certain cellular cells, cellular layer over here. This is known as nucellus. This is known as nucellus, and one cell among them form the whole structure like this. Okay, so you have the ovule within which you have the nucellus. Now outside the ovule you have two layers covering them. They are known as integuments. So it is situated. Okay, this embryo sac is situated inside the integument. Okay, inside the integument, that is fine. But outside the nucellus, is it outside the nucellus? No, it is inside the nucellus only. So this is a wrong statement. It has an egg apparatus situated at the chelazal end. Is the egg apparatus at the chelazal end? No, it is in the micropylar end. So what is the correct answer? A and B is correct statement. So I'll say C is the correct answer. Cool. So I'm not even breathing, I'm just teaching. So I hope you understand my pain and you also answer this. Okay, now identify the correct sequence of arrangement of cells in the mature embryo sac from the micropylar end. From the micropylar end we just saw, we have what? We have synergids over here, then we have the egg cell, then we have central cell, then we have antipodal cells. So let's see, synergids, then egg cell, then central cell, then antipodal cell. This becomes your answer, a very straight question. Okay, are you? Yes, cool. Now next question, even in the absence of pollinating agents, seed setting is assured in. Even in the absence of pollinating agents, that means you do not need butterfly, you do not need any pollinating agents, still you will be finding that seed is being formed. How can that be happening? Basically, when the flowers are clistogamous, the closed flowers with both the male and the female parts, right? Remember? So that is found in 3 types of flowers. I told you that is Viola, Oxalis and Commelina. So over here, where is Viola, Oxalis or Commelina? Only at one place, that is this. So here only you can find assured seed set, my dear children. Okay? Okay. Now, select the correct sequence of anther wall layers. Anther wall layers, I told you. So, in anther, you have sporogenous tissue. This, guarding it, you have what? Tapetum. Outside the tapetum, so this is tapetum, okay? Outside the tapetum, you have middle layer. Outside the middle layer, you have endothecium. So, I am writing endo. Outside it, you have epidermis. So, epidermis always comes outside. Middle layer is there and endo is coming inside, okay? So, between endo and the tapetum, or basically between these guys and the tapetum, you have the middle layer. You can remember like that. So, epidermis, endothecium, middle layer and tapetum, this becomes your answer. Cool. Cool. Chalo. Next, which of the following is not a correct statement? Placenta is located outside the ovarian cavity. So, if you see the ovary of the plant, okay, so this is the ovule. Now, over here, you would be having the placenta, which will be providing nourishment to the ovule, my dear children. So, placenta is located outside the ovarian cavity. No, it is inside the ovarian cavity. This is the ovarian cavity. So, this is what? Not a correct statement. First statement itself is not correct, my dear children. But let's see the other options. Central cell develops into PUC after fertilization. That is, central cell develops into primary endosperm cell after fertilization. Yes, it is correct. So, there are two things that is being formed in the ovule, okay. One is the male gamete fuses with the female gamete forming zygote. Now, to provide nourishment, who should be there? Endosperm should be there. Who forms it? The second male gamete. There are two male gametes going to come. Second male gamete fuses with the two polar nuclei of the central cell forming the PEC, that is primary endosperm cell after fertilization. This is correct. Now, orchid contains more than one ovule in an ovary. Yes, it definitely contains more than one ovule in an ovary. Now, egg apparatus consists of one egg cell and two synergid cell. Yes, I told you that, and it is found at micropellar end. So, this becomes your answer, my dear children. Placenta is located outside the ovarian cavity. Now, 11th question. Select, did I say 10 question? Okay, anyways. Select the correct sequence of events in microsporogenesis. What is the correct sequence of events? So, you know sporogenous tissue, I told you. Okay, now each cell of sporogenous tissue is going to act like MMC, microspore mother cell. And this microspore mother cell is going to undergo meiosis 1. So, meiosis happens in two stages. So, there is meiosis 1, my dear children, where it will divide into two cells, okay. And here it is diploid, 2n number of chromosome, that is double the amount of chromosome is there, just like any random cell of the plants. Now, this will undergo meiosis 1, here it reduces to half, that means it becomes haploid. From here, this cell will divide into 2, this cell will divide into 2 in meiosis 2. Now, you get 1, 2, 3, 4 cells. What are these 4 cells known as? They are known as microspore, 1, 2, 3, 4, tetrad. Okay, they are known as microspore tetrad. Now, they are together. You have to separate them. So, when you separate them, what do you get? You get microspores, which will be giving rise to pollen grains, my dear children. So, this is microsporogenesis event. So, this becomes your answer. Sporogenous tissue, microspore mother cell, microspore tetrad, and then microspores. Okay, first again is correct. And with this, we complete one chapter, that is sexual reproduction in flowering plants, my dear children, which carries, I think, 11 marks in your exam. Now, this is a schedule for PUC board math as well as physics as well as chemistry. So, in everywhere you will be finding model paper, you will be finding 1 marker, 2 marker, 3 marker, 5 marker questions, and also the passing package. So, be alerted about that. I can just show you, like I can zoom it and show you. This is for mathematics. You can take a screenshot if you want, but it's already been put in the WhatsApp group. So, if you have not joined that, you can join that as well. Now, this is for physics, okay. This would be for physics. Again, you can take a screenshot, and over here you have for chemistry as well. Cool. Okay, going ahead with the next chapter, that is human reproduction. Again, I think there are 11 questions, but I have put 10. Okay, let's start. Before that, I want you all to drink some water, because I am also going to drink some water. But anyways, see this question, and by that time, I am gonna come by drinking some water. Okay, so scrotum helps in maintaining the temperature of testis, which is, yes, which is dash lower than the normal body temperature. Scrotum help in maintaining the temperature of the testis, which is dash low. So, basically testis does not want to be in high temperature, it is in the body temperature. It prefers to be in 2 to 2.5 degrees Celsius lower than the normal body temperature, because testis has a male role to play, that is what? To produce these sperms, and sperm formation cannot happen at high temperature. So, basically I will say 2 to 2.5 is correct. So, these two options are wrong, eliminating these two options, okay. Now, can I cut? Yes, I can cut. Now, over here, I cut the wrong option. Please don't do this in your exam, okay. So, this can be done only by Jasmine ma'am when she is teaching, not by you guys. Now, yes. Now, each of the testes has about B compartments. So many compartments as in you're talking about testicular lobules. How many testicular lobules do you have, darlings? You have 250 testicular lobules. So again, this is correct, this is correct. Oh my God. Now, and measures 6 centimeter in length. What is C centimeter? What is the length of the testis? 4 to 5 centimeter in the length is the correct answer. So this becomes, B option becomes your correct answer, my dear children. Okay. Cool. Next question. The function of myometrium layer present in the uterine wall is to. So uterus has 3 walls. Outside is perimetrium, then you have myometrium, and then you have endometrium. The innermost layer, endometrium, is a glandular layer. The perimetrium is there for protection, and the myometrium, which is there in the middle, it is made up of smooth muscles, my dear children. What muscles? Smooth muscles. And smooth muscles do not listen to anyone, not even me. Yes. So over here, when I move like this, these are skeletal muscles. So smooth muscles are involuntary muscles. And why do you need smooth muscles in the uterus? Basically, when the baby has to move out, at this point of time, the uterus have to contract and relax. So obviously, you need smooth muscles up there. So for that, what do you have, my dear children? You have myometrium. So does it undergo cyclic changes? That is the role of endometrium. Does it exhibit strong uterine contraction during parturition? Yes. What do you mean by parturition? That is the expelling of the baby outside the body. Now, does it give protection to the uterus, my dear children? No, that is perimetrium. Does it help in implantation process? No, that is endometrium again. So B is your answer for this particular question. Okay, fine. Now going ahead, seminal plasma in human males is rich in. What do you mean by seminal plasma? So you know, in the case of human males, no, we have not me. Basically, males have 3 accessory glands. How many? 3 accessory glands, and these glands are responsible for providing nourishment to the sperms which are going to come. Okay, so basically I'll say the name of the 3 accessory glands which are there. One is seminal vesicle. Okay, the other one is prostate gland, and the other one is bulbourethral gland. So seminal vesicle, my dear children, gives or basically produces fructose so that the sperm has some energy to move around. Okay, now talking about the prostate gland produces calcium and certain enzymes as well. Now this calcium is very, very important for the process of fertilization to happen. If at all, it goes to the female reproductive tract. Now talking about bulbourethral or Cowper's gland, my dear children, it is responsible for lubrication so that the sperm can easily move around. Now over here, all the secretions together, I would call this as seminal plasma. I will call all the secretions together as seminal plasma. So if your seminal plasma is rich in what? Fructose and calcium, glucose and calcium, DNA and testosterone. You have fructose and calcium 3 being written over here. So this is what your answer. Cool. Chalo. Next, statement 1 says the process of release of sperms from the seminiferous tubules is spermatogenesis. So in the testis, there is testicular lobule. Now in the testicular lobule, you have so many 2 to 3 seminiferous tubules. This is where the sperm formation happens. So they are saying the process of release of sperm from the seminiferous tubule is known as spermatogenesis. But with what I have learned, spermatogenesis is the process of formation of sperm only. It is not the release of sperm and all. When you say release of sperm, you're talking about spermiogenesis. Sorry. You're talking about spermiation. Okay, spermiation, I always say, remember like that aviation, in aviation also things goes, right? Similarly here also release of sperms from the seminiferous tubules is spermiation. I know it's confusing, which is why I'm telling this, okay? Do not go around thinking about it a lot. So this is the answer. Okay, so this is a wrong statement. Now statement 2, the spermatids are transformed into spermatozoa. Spermatids, they are like kids, they are not mature enough. Now they are transformed to spermatozoa, they are mature. Okay, because basically spermatids are round in structure, my dear children. Spermatozoa are kind of elongated, much looking like sperms only. So spermatids have to be transported into spermatozoa, and this process is known as spermiogenesis. This is entirely correct. So I'll say statement 1 is false, statement 2 is correct. Statement 1 is incorrect, statement 2 is what? Correct, my dear children. Okay, got it? Next. To produce 2000 sperm and 400 ova, how many spermatogonia and oogonia are required? So I'll tell you one thing. From one spermatogonia, it undergoes meiosis. It has 2 cells. There is primary spermatocyte dividing into 2. That is secondary spermatocyte. Both of them giving rise to 2 spermatids each. So from one spermatogonia, if you see, you are getting 4 sperms. You are getting 4 sperms. So 2000 sperms are there. Okay, 2000 sperms are there. That means if they are coming in the form of 4, my dear children, how many spermatogonia will be there? 500 of them will be there. So you know this is an option. I'll cut these 2 options. Now talking about ova. So first, primary oogonia is going to divide into secondary primary oocyte is going to divide into secondary oocyte and also one cell, but this cell degenerates. This is known as polar body. So there is no use of this. Now again this guy, primary oocyte will also divide into two. Again one more polar body, that is secondary polar body, and over here this is the, okay, wait, just a second. Yeah, this would be the ovum. This would be what? Oh my god, I forgot. This would be the ovum right over here. You got it? So from one oogonia, how many ovum you are getting? You are getting only one, because you saw this, even if it had divided, then probably this also would have given 4. But over here, this is not happening like that. So it will be only 1. So if you have 400 oogonia, my dear children, all of them will be giving rise to 1 ova. So it will be 400 ova only. So your answer becomes 400 spermatogonia and 400 oogonia, my dear children. Okay. Just a second. Okay. Going ahead. Okay. Now, the release of sperms from the seminiferous tubules is called, I told you, it's what? Spermiation, aviation. Remember. Okay. What is the correct sequence of sperm formation, my dear children? So first of all, you have spermatogonia. Now some spermatogonia will divide into rapid cells, okay? They will undergo mitosis. Among them, one will be differentiated, okay? One will be differentiated, as in it will have a function to do. That is to form sperms. So one spermatogonia transforms into primary spermatocytes, my dear children. This primary spermatocyte divides into secondary spermatocyte. Spermatocyte, basically over here, primary divides into secondary. Secondary spermatocytes give rise to spermatids. Now spermatids would be forming the spermatozoa. So spermatogonia, then spermatocytes, then tits, that is spermatids, and then you have spermatozoa. This becomes your answer. Okay, cool. Chalo, next. Which among the following has 23 chromosomes? That means you are asked about haploid content of DNA. You are asked about haploid content of DNA, my dear children. So which among them has haploid content of DNA? Does spermatogonia have? No. Okay, no, it does not have. When it forms, when it divides, okay, forming, first one is primary spermatocyte. When it divides into secondary spermatocyte, that's when it completes first phase of meiosis. That's when it divides. So obviously spermatogonia has diploid. Zygote is made from male and the female gamete. So obviously N and N coming together, it becomes 2N again. Oogonia is again like spermatogonia only. So it will be having 2N. What about secondary oocyte, my dear children? It would be having haploid. Why? Because the primary oocyte divides into 2. One is the degenerate cell. Another one is what? One is the polar body. Another one is what? The secondary oocyte which is being produced. So over here obviously it is completing meiosis 1, and whenever someone completes meiosis 1, it is a reducing thing that happens. Reduction happens, okay? The number of chromosomes gets reduced into half. So this will be haploid. So I will say this is the answer for the question 8. Cool? Chalo. Next question. Can it not come here this side? Okay, anyways. Next, match the given in column 1 with those in column 2 and select the correct options given below. So proliferative phase, you know, menstrual phase has 3 different phases, or basically 4, okay? First phase is menstrual, where the blood flows down, okay? The blood flows down, that is when the endometrium lining breaks down, and this is known as menstruation. So I'll say C is with 1, breakdown of endometrial lining. Now the second phase which comes is that slowly this endometrium is gone because thinking that fertilization is not anyways happening. But still, does our body give up? No. Our body feels that yes, fertilization can happen. So I should be making back the endometrium. Now this phase is known as proliferative phase. And while the endometrium is being made back, the follicles are also going to get matured. So this is known as proliferative phase and also follicular phase. Either way, for proliferative phase and also follicular phase. Now secretory phase, my dear children. Okay, so the follicles have generated, they are mature, the endometrium is also forming. At one point the follicle will be breaking open and will be releasing the ova, that is known as ovulation. That is the ovulatory phase. Now strictly after the ovulatory phase, what happens? So the Graafian follicles is ruptured, right? Whatever is left behind, it is known as corpus luteum, and this corpus luteum releases hormones to make the endometrium line thicken very much. Why ma'am? Why all of a sudden? Because ovum is being released. Anytime fertilization can happen. So who is there to provide nourishment? Endometrium is being formed, but you need thickened endometrium, right? And for that, the corpus luteum, which is left back, okay, that is the Garfield follicle which is left back, it forms to corpus luteum and it secretes progesterone, which thickens the endometrium lining. So this is also known as luteal phase because corpus luteum is made, and also in this particular phase the endometrium is made so thick and it is going to release, okay, this endometrium which is there, it is going to secrete lot of glycogen and all, thinking that what if the embryo comes fertilized, or what if the zygote is being made. That is what I meant. So this is going with this. So A goes with 2, my dear children. A goes with 2. Proliferative goes with 2. So this is the only option that we have. Let's see. And B goes with 3 and C goes with 1. So this becomes your answer. Cool? Come on. Come this side. Yes, it is here. Okay, now next question. Which of the following hormones is produced only during pregnancy by the ovary? So during pregnancy, only one hormone is being released by the ovary. It's not like only one hormone. Basically, during pregnancy, only this particular hormone is released. That is what I meant. So it is none other than relaxin, my dear children. So what about the other options, ma'am? So estrogen, my dear children, is always released by ovary. It used to be released. Okay. Now talking about LH, it is being released by pituitary gland. Talking about HCG, it is released by placenta. It is a placental hormone. Human chorionic gonadotropin. Now relaxin is a hormone released by ovary. During pregnancy only the ovary releases this. At the time it does not. Okay. Now, but this, okay, so here we had 10 questions only, and with this we are completing 10 questions of human reproduction. So we are going chapter wise according to your NCERT. So if you want, you can open your NCERT as well, but I don't want you to do that. Be in my pace. Let's do it. So whenever you are not able to understand a question, at that point you have to stop and you can speed it down. Okay, that's the only thing we can do. Now, why is the AC not working? Everything happens in one go. What do you think is the reason for this? Now I think the AC will start working, and with that we are going to go ahead with the next question, that is decreased MMR, that is maternal mortality rate. Decreased, okay. What is it talking about? The maternal mortality rate is decreased, okay, and the infant mortality rate is also decreased. And increased people are there in the reproducible age, as in there are more people in the reproducible age. Now in a population, this will result in what? So if mothers are dying less because of proper care, okay, infants are also dying less because of proper care, and the people in the reproducible age, as in the people who have the ability to reproduce, they are increasing. That means, isn't the population increasing? Yes. So can I say increase in the growth rate is being seen over here? Yes. So this is again a direct question over here with A as the answer. Next, which of the following is an indicator of reproductively unhealthy society? Unhealthy, okay, you're talking about unhealthy. Increase in infant mortality rate. Obviously, this is an indicator. Who wants the babies to die? Obviously, this is an indicator, so I'll say A is an option, correct option, but let's see the other options, okay? Decline in maternal mortality rate, that's a good thing, so that is not unhealthy. Widespread awareness of contraceptive, that is also a good thing. Detection and cure of sexually transmitted diseases, that is also a good thing. So we are left with only one option, this becomes your answer. Cool. Next, amniocentesis is a medical procedure where some of the amniotic fluid of the developing fetus is drawn to analyze the fetal cells and dissolve substances. Didn't understand? So assume there's a baby here. Okay, you know, baby is covered within the amniotic amniotic cavity, and over here it has amniotic fluid. Okay. So what happens is that the baby cells know some of it sheds down over here. So when the doctor takes this particular fluid, the doctor is also taking certain cells of the baby. Now what is there in the cells? Cell will be having nucleus, nucleus will be having chromosomes. So what do people do with it is that they determine the sex of the child with the help of this, and when they get to know that this is a female child, they do female feticides, which is totally wrong. Why was this particular thing invented? Only for finding certain genetic disorders, my dear children, because you're talking about nucleus. Nucleus will have the genetic material, right? It is a chromosome. So over here, obviously you can find the genetic disorders. That was the actual use. So it was banned in India in order to solve female feticide. Which of the following highlight the importance of amniocentesis despite the ban? So even it's banned, still the importance is still there. For what? Identification of decline in sex ratio, what is it? Prevention of sexually transmitted diseases, nothing. Determination of biological sex of the unborn baby, no. how can that be important? Okay, the detection of genetic disorder, that is the correct answer for this particular question. D. Okay, now going ahead. Statement 1 says, lactational amenorrhea is the absence of menstruation during intense lactation. That means when the mother is breastfeeding, at this point of time, there won't be menstruation happening in her body. Okay, so what do you call this term as? You call this term as lactational amenorrhea, as in menstruation is not happening. Now this is highly reliable method of contraception. So people having sexual contact in this particular period, is it a very good way or reliable method of contraception? Can they be very sure that obviously fertilization would not be happening and baby would not be formed? My dear children, it's a very wrong thing to say that. This is not a highly reliable method. Not at all a high reliable method. So I will say 1 and 2. Basically 1 is correct, but statement 2 is what? Incorrect. Statement 1 is correct and statement 2 is what? Incorrect. It is not at all a high reliable method. Depending on the female to female, it can change. So lactational amenorrhea said that basically it's during 6 months, initial 6 months, that's when it happens. But for females, it can differ because depending on their body, what type of body they have. Okay, so this is wrong. Now, statement 1 says IUDs are ideal contraceptives for female who wants to delay pregnancy or space children. IUD means intrauterine devices like copper T or what do you call it, like lipase loop and things like that. So, are they ideal contraceptives for female who want to delay the pregnancy or space children? Yes, it is actually a good method. Now, statement 2 says IUDs are most widely accepted methods of contraception in India. Is that true? Yes, again, that is true. Now, IUDs cannot be performed without the help of the doctor or nurses. So yes, that is there, but these two statements are written as it is in your NCERT. Whenever you complete the whole paragraph of IUD, in the next page at the top you would be able to see it. So I will say statement 1 and statement 2, both of them are correct. Where is this? Yes, A. A is the correct answer for this. Okay, going ahead with the next question. Oral contraceptive pills mainly contain what? Oral contraceptive pills, as in if you have this particular pill, then probably in your body contraception, it is going to act like a contraceptive method. It is not allowing fertilization to happen. It is not going to allow that. It's not allowing the baby formation to happen over there. So what is this oral contraceptive pill rich in? It is rich in what? Progesterone or estrogen. Progesterone or estrogen, or what do you call progesterone? Progesterone or estrogen, either way. So progesterone or estrogen, my dear children. Now you might think, ma'am, both of these hormones are really important for ovulation and things like that. No, in general sense, they are being produced in females. Then how can they act like oral contraceptive pills? Basically, in our body, they are there in limited amount. Now, when you are increasing the amount, obviously they would be doing something else instead of doing the actual function. So over now, when it's there, when it is given to a female as a contraceptive method, my dear children, what happens is that this particular progesterone-estrogen combination would inhibit the process of ovulation. Ovulation is not going to happen. Apart from that, you know, uterus, the starting of the uterus has a region which is known as cervix, if you know. Okay, there will be mucus over here in the cervix and it's known as cervical plug, so that the sperm cannot get inside. So these are the things that happens because of oral contraceptive pills, and in this way the pregnancy is not being happening. Okay, so B is the correct answer for this. Luteinizing hormone has no role to play in this. Okay, please remember that. It's just these two hormones. Going ahead with the next question, select the incorrect statement regarding vasectomy. Vasectomy basically is a surgical method of contraception where what you're doing, you are going to cut down the vas deferens. You're going to cut down the vas deferens. So over here you have the testis. In the testis you have the testicular lobules and seminiferous tubules. They open into rated testis. From the rated testis they open into vasa efferentia. Then they form epididymis. Then they have vas deferens which carries the sperm. So over here in vasectomy the vas deferens is cut, my dear children, and it is being tied up. So no sperm will be there in the seminal fluid. That is true. What is seminal fluid? Basically, when the sperm as well as what do you call it, the accessory glands which are there, they are going to produce what? Seminal plasma. right? And the seminal plasma, when it comes along with sperm, you call it semen. So over here, there will be just the seminal fluid, but will there be sperm? No, because the sperm transport is being stopped over here. You get it? So no sperm will be there in the seminal fluid. That is absolutely correct. You are told to find the incorrect option. Now it is a surgical method of contraception. It is also correct. Vas differentia is cut and tied. This is also correct. No spermatogenesis. My dear children, if you cut this vas difference over here, okay, don't you think spermatogenesis will still be happening? Spermatogenesis happens in the testes. What did testes do wrong? Here anyway, spermatogenesis will be happening, my dear children. Okay, now the sperms which are being formed, they might just get degenerated. That's the only thing that will happen, but still sperms will be formed in their body. So no spermatogenesis is the incorrect thing that has to be said. Now, which of the following does not give the defined action of contraceptive? Okay, I'll drink little water. You also drink little water. After I have been holding since a long time. So with this, Jas ma'am is promoting the importance of water. I hope you are drinking enough. Now, yes. So which of them does not give the defined action of contraceptive? Hormonal method prevents or retards the entry of sperms, prevents ovulation and fertilization. That is correct. Hormonal method does that. When we are giving progesterone and things like that, at that time it is preventing ovulation and fertilization and everything. Vasectomy prevents spermatogenesis. I just told you now. Does it prevent spermatogenesis, my dear children? No. So this is a wrong statement, no? So I have to target it. Barrier method prevents fertilization. Yes. For example, barrier method as in you are creating a physical barrier between the sperm and the egg. For example, condom is acting as a barrier method. Now it will obviously prevent fertilization. If they both are not meeting, how can fertilization can happen? Now intrauterine devices increase phagocytosis of sperm. That means killing of the sperms. Does it happen? Yes. You have copper T. In copper T, the copper ions basically are responsible for all these functions. Okay, they basically would not be allowing the sperms to move around and things like that. So they suppress sperm motility and fertilizing capacity of sperm. That is absolutely true. This is the statement that is wrong. Okay, next. Which of the following are not completely curable disease? I will give you a clue, and in that clue you would not have to read all the options also, but you should be reading it anyways. All of the diseases starts with H. Not completely curable disease starts with H. Hepatitis B, genital herpes, so you can take H over here, and HIV infection, my dear children. Hepatitis B, please remember it's not A or anything else or C, it is B. Genital herpes and HIV infection are something that are sexually transmitted and that are not completely curable as well. So people who get it have it throughout their life. Okay, got it. Next. A specialized procedure to form an embryo in laboratory is. So in the laboratory only you are forming the embryo. What do you call this particular process as? Gamete intrafallopian transfer. That's where you transfer the gamete inside the fallopian tube. So basically the gamete is there, another gamete will come, embryo will form inside the body of the female. Now artificial insemination is basically when you are providing semen, that is the sperm. Artificially, okay, probably because the male is not able to ejaculate or the number of sperm is very less and things like that. Now intrauterine insemination, again it's like inside the uterus you are providing what this thing. So now over here, intracytoplasmic sperm injection, this is the answer. Why? Because what you are doing is that you are taking in the cytoplasm of the egg and you are injecting sperm there. Now what will happen when this thing is done? Obviously it will form zygote, and the zygote will be forming the embryo. So this is the answer for this question. Okay, now this question is not entirely correct, but yeah. And with this, we have completed reproductive health as well, and now we are going to the fourth chapter, that is principles of inheritance and variation. Over here you will be finding 11 questions. Now this chapter is very important, I hope you know that. Now a plant having the genotype AA, capital A small a, capital B small b, will produce dash kind of gametes, my dear children. What kind of gametes will it produce? So over here, how do you find the number of gametes? So there's a formula, 2n. So n is the number of heterozygotes which are present. So over here, this is a heterozygote because both the alleles are different. This is also a heterozygote because both the alleles are different. So can I replace n with 2? Yes. So 2 raised to the power 2 is basically 4. So it will be producing 4 type of gametes, my dear children. Now if the situation was like this, capital A, capital B. Now in this type of thing, how many gametes will be produced? Only one heterozygote is there. So 2 raised to the power 1. So it will be 2 gametes. Get it? So here 4 type of gametes will be produced. This is the answer. Next, in a monohybrid cross between 2 heterozygous individuals, percentage of pure homozygous individuals obtained in F1 generation will be. So in a monohybrid cross, there is only one character and you are crossing 2 heterozygous individuals. For example, capital T, small t, capital T, small t. So let's see how many percentage of pure homozygous individuals we have to find. So capital T, small t, capital T, small t, this and this fuses, you get capital T, this and this fuses, you get capital T, small t, this and this fuses, you get capital T, small t, this and this fuses, you get small t, small t. Over here, what is the percentage of pure homozygous? Tell me. Who all are homozygous? This is homozygous, both the alleles are same. This is homozygous, both the alleles are same. So 25%, 25%, 25%, 25%. So if I say 25% and 25%, I say 50% of them are pure homozygous. This becomes your answer. Okay, chalo. Next question. Which of the following assertions about alpha thalassemia is incorrect? Incorrect. So it involves the HbA1 and HbA2 genes. Is it correct? Yes. So in alpha thalassemia, defects can be in the alleles of HbA1 gene or in the HbA2 gene. So this is correct. Is associated with the mutation or deletion of genes on chromosome number 11. So where are these HbA1, HbA2 genes present? On chromosome number 11 or 16? It is present on the 11th chromosome, my dear children. Now it is associated with mutation or deletion of chromosome number 16? No. Then if you are talking about 16, you are talking about beta chain. This is alpha thalassemia, that means your alpha globin chains are affected over here, not beta chains. So beta genes are present over here, so this is wrong statement. 16, it is beta. Now over here, how can you remember it? Now 16, if you see, my dear children, it looks something like B. So you can remember like that. How's the trick? Now, it leads to reduction in the synthesis of globin chain. Is it true? Yes, obviously. When these genes are affected, it leads to reduction in synthesis of globin chain. If it was written beta globin chain, then it would have been wrong. Here it is just globin, so it can be either alpha or beta. So it can be correct. So this statement is, my dear children, an incorrect statement. Rest all of them are correct. Okay, now which 3 scientists independently rediscovered Mendel's work? So Mendel's work was done, okay, by 1856 to 1863, he did his work. And then lots of people forgot his work. But there were 3 scientists who came up and said that we would be working on it. Who were those 3 scientists? Their name is direct, my dear children. There is no need to explain. Dave, Rice, Correns and Schemark. Dave, Rice, Correns and Schemark were the people who rediscovered Mendel's work independently. So this becomes your answer. Okay, now next, what is a possible blood group of an offspring when both the parents have AB blood group? Suppose both the parents AB, AB. What are the possibilities? Without even doing, I can tell. Can you tell? So basically, AB can give A gene, B gene. Sorry, A allele, B allele. A allele, B allele. This and this fuses, you get AA. That means A blood group is there. This and this fuses, you get AB. Both are the dominant, co-dominant, so AB blood group. This and this fuses, you get AB again. This and this fuses, you get BB again. That means it is B blood group. So A blood group, AB blood group and B blood group is seen over here. So B is your answer. Okay, cool. Chalo, next. Which of the following is incorrect regarding ZWZZ type of sex determination? So ZWZZ type of sex determination is found in birds. Is it found in birds? Yes, it is definitely found in birds. Females are homogametic and males are heterogametic. Do you think the females are this? And the females are this and the males are this? No, my dear children, it's opposite. Females are heterogametic. Okay, females are the one that are heterogametic. So they will be producing 2 type of gametes, either Z or W. Okay, usually it's the male who are like that, but in the case of birds, females are heterogametic and the males are homogametic. So this statement which is there, this incorrect statement, and we had to find what the incorrect statement. But let's see the other option. Total number of chromosomes is same in both male and female. That is true. So this is wrong. Okay, this is an example of female heterogameity that is in birds. Going ahead. Haplodiploid sex determination is found in. Haplodiploid as in, if a cell is diploid, that means it has 2 N, it will be a female. If a cell is haploid, that is, half the number of chromosomes than it will be a male. This type of sex determination is seen in what? Grasshopper, birds, humans and honeybees. Honeybees are like that, okay? They show haploid diploid sex determination and it is the only examine given in your book. So you can remember like that. Going ahead, next option. So next question. In XO type of sex determination, females produce two different type of gametes. In XO type of sex determination, is it females who produce two type of gametes? No. Females will be XX. So they will be producing only one type of gametes. The gametes that has X chromosome. But XO will be produced by males. This is again an example of male heterogamety. Male hetero. This is not female hetero, male hetero. So female produce two different type of gametes. This is wrong. Males produce two different type of gametes showing homogamety. So people will do one thing. No, males produce two type of gametes. Yes, correct. That is wrong. You have to read. Homogamety is being mentioned over here. Who will read that? So females produce gametes with X chromosome. That is totally wrong. Now males produce two different type of gametes showing heterogamety. Yes, that is correct. So read your questions properly before answering them. Okay. Next, when is it possible for a woman to be colorblind? So listen, color blindness is a sex-linked recessive disease. That means the woman would not be affected if one of her chromosome is affected. She will be just a carrier. Now if I want her to be colorblind, both X should be affected. Both the X chromosomes should be affected. Now when will that happen? That will happen when the mother is a carrier and the father for sure he is colorblind, because father is going to contribute one X chromosome only. If that X chromosome is normal, then she would also be a carrier. So listen, this is the condition in that case. When this and this comes together, you get colorblind girl child. But suppose the father is not having color blindness, then the X, whatever X will come from this side, it will be safe option, right? So always it will be like this, and if this comes from the mother, then she again will be a carrier, she won't be affected. Now if you want a woman to be colorblind, my dear children, the father cannot have a normal vision. Listen to me very carefully. If you want a woman to be colorblind, the father cannot have a normal vision. So wherever you see normal vision, cut that option. A father is colorblind and the mother has a normal vision. Yes, she can have a normal vision, right? She is just a carrier, so this can be true. Now father is colorblind and mother is a carrier. Oh sorry, normal vision, sorry, this is wrong, this is also wrong, okay? Because why? Because mother cannot, basically she has a normal vision, but over here there is something more specific is given, that is she is a carrier. So carrier, do they have normal vision, my dear children? Yes, they have normal vision only. So this statement is also not false, but over here there is more specific statement, that is the father is colorblind and the mother is a carrier. In that case, definitely. Now normal vision can be like this also, right? Normal vision can be like this also, if you do not want that option to be there. So this is the correct answer. Okay? Ouch. Okay, I got myself hit. Yes. And 10th question. If both the parents are unaffected carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? So both the parents, my dear children, so I am just saying, are unaffected carriers. So they are carriers, as in I am just writing T over here. So this is the mutated allele, okay, gene mutated allele. Yeah, this is the mutated allele. Now over here another parent with another mutated allele. That is, it is recessive disorder, my dear children, right? So obviously it should be in this condition. Recessive will be in this condition. And whenever you say your carrying for recessive, that means it should be this condition. Why? Because this is dominant, which is why this is not being expressed. That is why they are carriers. If suppose both the alleles were like this, that's when you say they show the disease. And why does this happen? Because this is a recessive disease, and recessive condition only shows up when you have two alleles of the same kind. Now you are asked, what are the chances of pregnancy resulting in affected child? So let's see. So this can be an allele. So this can be in a gamete. This can be in a gamete. Now this can be in a gamete. Now again, this can be in a gamete. Suppose this and this fuses, you get HB and HB. This is normal child. This and this fuses, you get HB and HBT. Now this is a carrier. Now you fuse this with this, again HB and HBT. This is also a carrier. You fuse this with this, you get HBT and HBT. Now this is the disease. So 25%, 25, 25, 25. So what percentage of the chances that the child will be affected? 25% chance are there. Got it? So this becomes your answer. Going ahead. In the F2 generation of the Mendelian dihybrid cross, the number of phenotypes and genotypes are. So you might be aware about the 9:3:3:1 ratio, right? 9:3:3:1 ratio. So 9 represents there are round 9 of them who are round yellow. One of them is wrinkled and green. That is the recessive trait. Wrinkled and green. 3 of them are the new combination, that is round and green. And 3 of them again, they are wrinkled and yellow. So wrinkled and yellow, green was an option, but this is wrinkled and yellow. So this is a phenotype, this is a physical appearance, this is also physical appearance. 1, 2, 3, 4. 4 phenotypes are being observed. 4 type of phenotypes have been observed. What about the genotypes? So my dear children, the genotypes as a genetic makeup, there are 9 of them. There are 9 of them. So you have to remember it like that. So phenotypes are 4 and genotypes are 9. So if you want to see the genotypes, these are the genotypes. It can be capital R, capital R, capital Y, capital Y. It can be capital R, capital R, capital Y, small y, and things like that. So there are 9 of them. So 9 genotypes and 4 phenotypes. Please remember, it's a very important question. Now, one very important question which I have not included over here. Okay, that is whenever you say that basically 2 genes are there. In Drosophila experiment, 2 genes are there and they are showing high amount of recombination. They are showing what? High recombination. What does that mean? That means, my dear children, the genes which are there, they are far away from each other, which is why they show high amount of recombination. That means this part of the chromosome will be moving to the other part and forming a new part over there. You get it? There is a new combination that, and whatever part was over here, it will be coming over here. So you find a new recombination that is happening. When does new combination happens? When the genes are loosely linked. When the genes are loosely linked. Now on the other hand, if you said that the amount of recombination is less, that means the chromosomes, the genes are closely linked. They are linked genes or they are closely linked. So if at all I cut this portion and I put it there, still these genes are together. So is there a chance for recombination to happen? No, which is why there is less recombination. So I will say it is closely linked. Always remember. So if it is said that 37.3% recombination is seen, that means it is too much recombination. 50% is the highest amount of recombination. So 37.3, that means high recombination is happening. That means they are far away from each other. The distance is really far and they are loosely linked. If it is said that 1.something% is recombination that is being observed over here, that means they are closely linked. The distance between the genes is so less. Okay, please know this concept because you might get questions from this concept as well. And with this, we are completing principles of inheritance and variation as well. Now, molecular basis of inheritance, my dear children, you have 15 questions. So, are you ready? Already it's been 56 minutes, but it's okay. I hope it is helping you well. Isn't it helping you well? So, do let me know in the comment section, my dear children. Also, if you have any doubts until now, you can put the name of the chapter and the question number. Okay, in that way, I would be able to understand what is your doubt. Works for you? Then it works for me. I hope every one of you. So listen, while I am solving the question, there might be a lot of you who might be thinking that I do not know the answers. What kind of person I am? I am not prepared and all. So don't think like that. You are preparing now. Do you have any other option now? You cannot go to the back and change anything, right? You have now and you can bring changes over here. So we are bringing the changes over here. We are solving questions from molecular basis of inheritance. First question. The 2 nucleotides of DNA are linked through 3 and 5 carbon of pentose sugar by. So, my dear children, this is a pentose sugar, suppose. 1, 2, 3, 4, 5th carbon is like this. Now, 5th carbon is attached to phosphate. Now, this phosphate, it is attached to 1, 2, 3, 3rd carbon of another nucleotide. Now what do you call this bond? That is what is being asked. Two nucleotides of DNA are linked to 3 and 5 carbon of pentose sugar. By what bond? So this bond which is coming over here, this is known as phosphoester bond. Ester bond, this is ester bond, okay. This is again phosphoester bond. When two ester bonds are there, what do you call it? You call it phosphodiester bond. You call it phosphodiester bond. If you are asked what is the bond between the 5th carbon and the phosphate group, then it is just phosphoester bond. Since you are asked between 3 and 5, that means you are including 2 ester bonds, so it will be phosphodiester bond, my dear children. Do not get it wrong. I'm going to kill you if you do that. Now going ahead. In biochemical characterization of transforming principle, which of the following enzymes affect transformation? So, transformation, basically what was happening is that the genetic material, no one knew what was the genetic material, okay. So, they were saying that basically in an experiment, there was the dead S strain, okay, I am writing dead spelling like DED, oh my god, okay. There was dead S strain. Now, S strain is a virulent strain, my dear children. Usually when S strain is live, it causes disease, but this is dead S strain. Now, when you are mixing it with live S strain, live R strain, which was actually non-virulent, you found that basically some things are moving from here to here and making the R strain convert into live S strain. So, there was some transformation happening over here. So, now you are asked, which of the enzymes affect transformation? Which of them does not allow this transformation to take place? So, people try to, that is what was the name? Avery, MacLeod, McCarthy, all of them, they tried lots of things, okay, lots of enzymes. So, first thing they treated with protease, that means the one that digests the proteins. So, when they did that, the proteins were killed, but still transformation was taking place, okay. Then they tried with amylase, that means it was dealing with carbohydrates. So, carbohydrates were broken down, still transformation was taking place. Now, they treated with lipase. When lipase, that means fats are being broken down, still transformation was taking place. But when they treated with DNAs, my dear children, now in this DNA is getting damaged. Now when DNA is getting damaged, you didn't find this transformation happening. So what is that enzyme that affects transformation? It is DNAs, and that's how they got to know that DNA is the genetic material that is probably getting transformed from here to here. You get it? But still people didn't believe it until Hershey and Chase did their experiment, okay. So yes, this is the answer. Now, the experiment that proves the semi-conservative principle of DNA replication. So, DNA replication, the semi-conservative principle of it was proven by Michelson and Stahl. So, this is a direct question, and they did the experiment on E. coli. Okay, they did the experiment on E. coli. Now, RNA polymerase 1 transcribes. So, you know in eukaryotes we have 3 RNA polymerases, RNA polymerase 1, 2 and 3. So, 1 basically helps in the formation of ribosomes. So, we have to look for rRNAs, ribosomal RNAs. So, RNA polymerase 1 helps in the formation of ribosome by making rRNA, because rRNA is a major constituent of ribosome. So, this becomes your answer. But to confuse you, you are given 5S rRNA and your tRNA, hnRNA, things like that. No, the answer is what? rRNA, that is made by RNA polymerase 1. Now, RNA polymerase 2, my dear children, is responsible for hnRNA. Yes, it is responsible for formation of hnRNA, and RNA polymerase 3 is responsible for tRNA as well as 5S rRNA. Got it? So, 1 is for rRNA, always remember, ribosome, R for RNA and R for RNA and R for ribosome. My tongue is getting twisted. Okay. Now, GGU, GGC, GGA and GGG all code for glycine. All of them are coding for glycine. Which salient feature of the genetic code does it prove? So, in the genetic code, when there are multiple codes for single amino acid, you call this feature of the codons as degenerate feature, or you call this codons as degenerate codons, as in you think you have only, your best friend has only you as best friend, but your best friends have multiple best friends. So, basically it's kind of confusing, but you can remember like that. Basically, for one particular amino acid, you have multiple codons, my dear children. That means these codons are degenerate codons. Okay? Going ahead. Amino acylation of tRNA occurs during amino acylation of tRNA, as in, you know, tRNA reads the mRNA. tRNA translates the information that is there in the mRNA to form proteins. Now what does tRNA do? tRNA basically brings amino acids with it. And this process where the amino acid comes and joins to the tRNA, my dear children, initially before it comes over here, it comes like that only. Okay, it has one amino acid attached to it. Now this process is known as amino acylation. Amino acylation. So where is this happening? Amino acylation of tRNA occurs during what? The process of translation, where the mRNA is getting translated. So your answer is this. Okay, now amino acylation of tRNA is also known as charging of tRNA, and this is highly energy required process. Okay, it needs high amount of energy. Going ahead, in a single stranded DNA, if the ratio of A and G, A raised to G is what? 60 by 40. Adenine this much is guanine is this much. Then the ratio of T and C is so you might think, ma'am, 60 adenine is there, obviously it makes base pairs with T, so it should also be 60 and this should be 40 again. But my dear children, did you notice the question? It is asking for single stranded DNA. If this is the strand of DNA, over here someone is saying that adenine is 60. Okay, sorry, not 600, it is 60 and guanine is 40. Does it matter? Thymine can be 60 or 70 or 80, whatever is there. Does it matter? You are talking about the base pair. You are not talking about the base pair, my dear children. You are talking about a single stranded DNA. So none of the above becomes the answer. Most of the chance, whenever none of the above and all of the above options are there, okay, there are higher chances that it is correct. So none of the above is the correct answer. And now, if a mRNA, this is a very interesting question. If a mRNA, so I did these questions in Chakravyuh series, and you might be thinking that, ma'am, it's basically repeating questions. These questions are important, so shall I not repeat? So people who didn't see through the Chakravyuh series, should I not do anything for them? What are you saying? So basically, it's for you to, now you can write it out still, and I am pretty sure you might be getting it wrong as well, because it happens. Okay, so if a mRNA, because you have a lot to study and I get you, it's totally fine, but this is how we learn. So if a mRNA, that is 3-AUGUUUUAA 5- undergoes translation, how many types of amino acids are present? So what you do, I'll tell you. Okay, so you'll say AUG is there, this will code for methionine 1, UUU for 1, phenylalanine, this also 1, this is also 1, UAA stop codon, so does not code for anything. Basically 4 types are being formed. Is it true? Is it the correct one? First thing that you need to know is mRNA is read from 5- to 3- end. It is read from 5- to 3- end. Right. Now, how many types of amino acids is being formed is asked. So, AAU will be forming 1. Okay. UUU will be forming phenylalanine. This also forms phenylalanine. This also forms phenylalanine. So, I will say 1 type of amino acid, right? Phenylalanine is same thing, so 1. Now over here, GUA will be again coding for another 1. So 1 plus 1 plus 1, you have 3 different types of amino acids being made up over here. Got it? I hope you got it. Fine. Chalo. Next, which of these are correct? UTR, untranslated regions, are located on DNA, is it? Untranslated regions, my dear children, who undergoes translation? mRNA. So, untranslated region will be there in the mRNA, not in the DNA. So, I will say this is wrong. Now, UTR is present both at the 5- before the start codon and also at the 3- after the stop codon. Yes. So, in the mRNA, before the 5- start codon, there will be untranslated regions. After this stop codon also, there will be untranslated regions. So, this is correct. Now, UTR, what is the use of it? It provides stability to the mRNA. This is also correct. Now, UTR is required for efficient translation. That is also correct. So, I'll say 2, 3 and 4 is correct. So, 2, 3 and 4 is correct. That means D option is entirely correct, my dear children. Okay. Yes. So, this is a 2-mark question also. You will be asked about UTR. Define UTR. So, you should be knowing UTR. These are the points that you have to write. Next. Splice, methyl guanosine triphosphate is added at the 5- end of hnRNA in a process of. So, what do you mean by hnRNA, my dear children? So, when I say that DNA forms mRNA, it's not a direct process. When I say transcription is happening, basically it's not mRNA that is being formed. It is hnRNA that is being formed first. This hnRNA will have to undergo certain processing to form the mRNA. Okay. So, over here, first process that is there is capping, where methyl guanosine triphosphate is added at the 5' end of the hnRNA. Why is it done? Because you want to protect the hnRNA, or basically the future mRNA, from the action of certain enzymes and all. That is why you are doing the capping process. Okay, so methyl guanosine triphosphate is added at the 5' end, and this is known as capping. Now at the 3' end you add lots and lots of adenine over there, and this is known as tailing. Okay, this is known as tailing. Now one more thing, in hnRNA you have this two regions, exons, introns, exons. Exons are the coding regions and introns are the non-coding regions. They don't code for anything. So you remove them, and this process is known as splicing. So after doing all this thing, you finally get the mRNA. So over here, methyl guanosine triphosphate is like a cap. So this is the capping, and this is the answer. And let's go with the next question. Human insulin is produced in E. coli cells by rDNA technology. This proves the genetic code is. So human insulin in E. coli, that is a bacteria. How can that be produced? Basically, you're putting the gene over there in the bacteria, and the bacteria is still being producing that protein. This proves the genetic codon is what? Universal. It is the same. So whatever you in my body and you in E. coli's body will also produce what? Phenylalanine only. So I'll say this is what? Universal. This proves the genetic code is what? Universal. Cool? Going ahead. Question number 12. If A and B are the DNA of two mothers, this and this, okay, are the DNA of two mothers, and C is the DNA of the child, okay, the conclusion based on the banding pattern of DNA fingerprinting can be drawn as. So if we see the banding pattern over here as well as over here is the same. So this type of banding pattern can be similar only in mother and child. So I'll say C is the daughter of B. C is basically daughter of B. So child C belongs to mother A. Ayo, don't give. Child C belongs to mother B. Yes, that is the correct answer. A and B are sisters. No. No conclusion can be drawn. No. This is the answer. Okay. So just look for the banding patterns and just tell. And suppose it's not mother and child question given. This is the culprits. Okay, this is sample from the murder site, and these are the culprits with culprit 1, culprit 2. So this culprit 2 who did the crime. This type of questions can also come. Just understand it. Now 13. Match the following. Alec Jeffrey. What did he give? F. Sanger, Frederick Sanger, what did he give? Jacob and Monod, what did he give? Griffith, what did he did? So basically, first do the questions that you already know. Griffith, you might be knowing that he did the transforming principle experiment with that mice and all. So D goes with 2. Let's see in which option D is going with 2. So there is only one option where D is going with 2. Rest all the options I can cut down. Now let's see. A is 4. Alec Jeffrey, DNA fingerprinting. Is it correct? Okay, let's see. B. F. Sanger, that is Frederick Sanger, gave the annotated DNA sequencing technique. Is that true? Yes. Okay, now talking about Jacob and Monod, they gave the Lac-Operon. Yes, that is also coming true. So this becomes your answer. So please remember, my dear children, Alec Jeffrey was the person for DNA fingerprinting, F. Sanger is for annotated DNA sequencing method, and your Jacob and Monod for Lac-Operon. Ayo, why did I mention D? D is 1. So this is a wrong thing, okay? I have mentioned the wrong thing. This is the correct answer. Don't get it wrong. Now, question number 14. Statement 1 says deoxyribonucleotide triphosphates, DNTPs, act as substrate during DNA replication. Suppose DNA is this is DNA with ATGC, ATGC. So over here, the nucleotides which are coming here, no, they are known as deoxyribonucleotide triphosphates. So what is it? It looks like this. So over here, 1, 2, 3, 4 and 5th carbon. On the 5th carbon there is phosphate group, but actually it's not just one. There are 1, 2, 3 phosphates. It's triphosphate which is there. Now when this nucleotide is being added, the next nucleotide comes and it will be binding on the first phosphate, leaving the other two guys. So over here, when the phosphate bond breaks, no, there is too much amount of energy, and this energy is utilized for making of another bond with the next nucleotide. So the substrate is definitely substrate as in the one that can be used for formation of something. So deoxyribonucleotide triphosphates, DNTPs, are substrate during DNA replication. Yes, they are substrate. With the help of them only I can make this. Now deoxyribonucleotide triphosphates are also acting as source of energy. Yes, they are acting as source of energy. Why? Because when there is a phosphate group, 3 phosphate group, when I am binding with one phosphate, I am basically breaking this bond, and this bond will be giving me lots of energy, my dear children. So your answer becomes what? Both the statements over here are completely true. Both A and B are, statement 1 and 2 are correct. This one. Okay. Now last question from this chapter. In pea plants, each character is governed by a gene which is. In pea plants, each character is governed by a gene which is. So in pea plant, pea is a plant. Plant is what? A eukaryote, my dear children. And eukaryotes have monocistronic, genes. Okay, they are monocistronic. What do you mean by monocistronic? As in one cistron, my dear children, will be having only one gene coding for one particular protein. Not like prokaryotes. Prokaryotes, no, they are polycistronic. That means in one they will be having multiple genes coding for multiple proteins. So they just code for one protein, which is why they are monocistronic. Eukaryotes, V plants, all are eukaryotes. Okay. Now one more concept, lac operon concept. So you should be knowing one thing. In the lac operon, my dear children, over here always a gene will be having a promoter sequence. Now suppose there is a gene that will be coding, there's a regulatory gene over here. This regulatory gene has its promoter sequence. Now there will be another promoter sequence for another gene, but there will be an operator region also here, suppose. And these are the genes Z, Y and A. Z codes for beta galactosidase, okay, and Y codes for permease and A codes for transacetylase. Now listen to me. Lactose, when it's there, this is about E. coli. E. coli has it, okay. Lactose, when it's there, beta galactosidase will be breaking down this lactose into smaller units and will be utilizing it for energy. Permease, which is there, it is allowing the lactose to get inside by making the membrane permeable. Transacetylase has some other function. So this is the normal functioning, okay. But we do not want these enzymes to be made unless and until lactose is present. So what do? This regulatory protein, regulatory gene codes for a repressor protein. Repressor protein. This repressor protein will be going and binding on the operator. Now if there is something bounded over here, even though the RNA polymerase comes and binds on the promoter region, it cannot be moving ahead. And hence transcription would not be taking place, hence formation of enzymes would not be taking place. But if lactose is present, this lactose guy becomes strength with this, and hence this does not have an option to go and bind over there. Now the RNA polymerase can do the polymerization reaction. So this is lac operon, my dear children, and please remember it is going to act like a negative thing. Okay, it's mostly in the negative state. It's a switched off state. In the switched off state, lac operon is a switched off state. Okay, it is not going to do the process of transcription unless and until lactose or allolactose is there. Okay, and with this we cover that and we get into the next thing, that is evolution, my dear children. So evolution, I have added 11 questions, so let's do it. What's the time now? 1 hour 15 minutes. Okay, so theory of spontaneous generation was disapproved by experiments conducted by. So theory of spontaneous generation, as in, for example, there was a sac of something, and from the sac mouse was coming out. So people were thinking, oh, from the sac spontaneously things are getting regenerated. No, it was not like that. So who disapproved it? It was Louis Pasteur, my dear children. It was Louis Pasteur. What did he do? He had a flask, okay, he had a very closed flask, and he had killed yeast inside that. And he found that there is no growth, extra growth. But when he kept this flask open, air was getting in and there were certain changes found over there. So that means air had some things. So there are organisms around in the environment which leads to formation of something else. So Louis Pasteur was the one who disapproved the theory of spontaneous generation. Okay. Next question. The thorns of bougainvillea and tendrils of cucurbitae represent what? Homology, convergent evolution, divergent evolution, or analogy. So basically, suppose this is the plant of bougainvillea. Over here, the axillary bud will be giving rise to thorn, which provides some protection. Now, suppose this is the plant of cucurbita. Over here, the axillary buds give rise to tendrils. So, if we see, they are from a common ancestor. They are from a common ancestor, okay, and the structure is also same. The internal structure, if you see, structurally also they would be same, but the functions they are going to perform is totally going to be different. So, this type of structures are known as homologous structures. Homologous structures, and since they are in different habitat, they are diverging. So this is an example of divergent evolution. Divergent evolution. So I have to look for homology and divergent evolution. So I'll say 1 and 3 is the correct option. 1 and 3 are correct. Okay, so even the structure is not same, ma'am. Internal structure, as in it is both coming from the apical, from the, what do you call it? axillary bud, right? Both of them are coming from the axillary bud, which is why I said structurally it is same. Not that the tendrils is looking like a thorn. Don't go like that. Okay, fine. So yes. Next, Charles Darwin in Galapagos Island found that finches have different forms of beaks. This process of evolution is called as. So basically what happened was, suppose this is geographical area, in between you had finches. Now finches will be having mutation, obviously, just like other organism. We all have mutation, they also had mutation. Some's beak were like this and that. So some of them, when they moved over here, they were able to better adapt in this particular area. Some when they moved over there, they were able to adapt over there better. Similarly, they went all around the area in the Galapagos Island, and all of them had different beaks because of the mutation that might have already happened. Now with the help of these beaks, they were able to eat seed, or basically insect. They were able to eat grass, lot of things, different things. So this type of evolution, my dear children, this process of evolution is known as adaptive radiation. So they are radiating to all the areas and they are getting adapted over there. So your answer becomes B. Cool. Next, a brown-billed duck is newly added to a farm of white-billed duck. This indicates what? Brown-billed duck is newly added. So over here, first we had a population of white ones. So genetic frequency of white ones were more. Okay, white. Now I am adding something new. The frequency get disturbed. Now I am adding brown. So over here, am I not bringing something in? A new gene. So I will call this as a gene flow. I will call this as gene flow or gene migration, my dear children. So your A becomes the correct answer. A brown-billed duck is newly added to a farm of white-billed duck. This indicates gene flow, not genetic drift, not mutation, not natural selection. Mutation as in, if some mutation is happening over here, then it's okay, but you're introducing something, right? So it is not mutation. Genetic drift as in it is occurring by chance. Here it is not occurring by chance. For example, I came in, I stamped over them and I killed all the white ones. So there's a genetic drift that is happening. The allelic frequency is disturbed because of a very random process. Now natural selection is when nature is selecting. Here nature has no role to play. Okay, so your answer is gene flow. Now, all these things, my dear children, affects the Hardy-Weinberg's principle. So basically they put down that the genetic frequency or the alleles frequency will be always remaining same all throughout the generation after generation, but it's not like that. It changes. Okay. Now, the biggest land reptiles that existed during Mesozoic era was, that means the reptilian era. Okay, so over here, what was the biggest land reptile which was there? A big form of dinosaur, that is Tyrannosaurus, my dear children. It is what? Tyrannosaurus. Please remember it, very important. Next, arrange the following in the order of evolution. So first came, okay, we had two things, Dryopithecus and Ramapithecus. Dryopithecus were more ape-like, Ramapithecus were man-like, Ram. You can remember like that. From Ramapithecus, that is from C. So C is the first thing that should be the Dryopithecus, anyways is not mentioned. So C should be the one. So over here all these options is wrong. Why? Because none of them are starting with C. So you have Ramapithecus, and from Ramapithecus you had Australopithecus. C then B. From Australopithecus you have the first human-like, okay, or properly ape-like, sorry, not ape-like, human-like thing, that is Habibi, Yella Habibi, Homo habilis. Okay, so this becomes the answer. And then Erectus, Homo erectus, they were able to stand erect and things like that. And then came Neanderthal man, my dear children, so this becomes the answer. Okay, so always remember Ramapithecus first, then Australopithecus, then Habibi, Homo habilis, then Homo erectus. From Homo erectus came Neanderthal man and also the modern day Homo sapiens. Okay. Next, in Miller's experiment, the environment of primitive earth was created and the temperature was. So in the primitive earth, the temperature was really high, my dear children. So when Miller created an environment similar to that, he made sure the temperature was high and it was 800 degrees Celsius that he chose. Always remember, 800 degrees Celsius, and it was a reducing environment. That means there was no oxygen up there. These two criteria have to be remembered. Mostly straight. I told you the answer already. I'm so sorry. So the paleontological evidence for evolution is obtained from paleontologically, as in you're talking about the fossils and everything. So this type of evidences of evolution comes from the fossils. So when you go around the different rocks and things like that, you find organisms, fossils, and then you get to know that, oh, before these many years, probably they existed in this earth. Get it? So that is the paleontological evidence which you get from the fossils itself. I showed you the answer. Should we cancel this question? But okay. Now, embryological evidence for evolution was disapproved by. I'm not asking who gave it. Who gave embryological evidence? It was Ernst Haeckel. What do you mean by embryological evidence? That is, for example, I am a human being. So when I was an embryo, I was inside my mother's womb at this point of time, I had a small tail. Okay, I had slits, gill slits. Okay, lot of things. But when I was out as a baby, did I have all that? No. So, embryological evidence as in, probably I and you and every one of us might have evolved from fishes. Then from fishes you got, what say, amphibians. That is the one that live both on land and water. Then from them you got reptiles, and then from them birds and us. Okay. So this thing that is what say a function of evolution is seen in our body also during what? During the whole process of embryogenesis, when we are embryo. So that was embryological evidence of evolution. So Ernst Haeckel said, since we are evolving like this, probably this might be the correct way of evolution. But there was someone who said that this is entirely wrong, and that was, so the same name the person is there, it is Carl Ernst von Baer. Carl Ernst von Baer, he said that I won't bear all this thing. You can remember like that, okay? Carl Ernst von Baer said, I won't bear all this thing. This is completely wrong. In that case, it should show reptiles features also. It should show other features also. How can it just show a tail and a gill slit? So that was how he said. Not exactly how he said, but yes. Okay, so I hope you remember in this way when I teach in this way, and I hope you're not getting bored when I teach. Okay, now which is of the pair? Okay, which of the pair is neither homologous or analogous? So homologous as in they should be having different functions, but be coming from the common ancestor. For example, tendrils of cucurbita and thorns of bougainvillea. Okay, so this is homologous, my dear children. So you had to find what is either homologous or analogous, but I'll tell you about analogous also. Analogous as in they have a common function, but structurally they are not same. For example, wings of butterfly and wings of bird, both have wings for flying, but is the structure same? No. They have common ancestor? No. Okay, but they have evolved, they have converged because they are living in the same habitat, that is air. So they show analogous organs, that means they show convergent evolution. They do not have a common ancestor, but same function will be there. Homologous means opposite of it. They will be having no common function, okay, but the structure would be same and the common ancestor would be there. So here only one thing is mentioned. Sweet potato and tendrils of cucurbita is not the example. Is sweet potato and potato was given, that would have been the correct answer for analogous organs, because sweet potato and potato, both of them are storing things. So function is same, but are they arising from the same structure? No. Sweet potato is a root modification and potato is a stem modification. So they have different ancestor or different structures internally, but common function. So that is an example of analogous thing, but that is not given over here. Okay, chalo. Oh. Next, 11th question. Observe the following diagrammatic representation of operation of natural selection of different traits. Here, natural selection depicts. So basically, nature is selecting something, not something. It is selecting two things. That is why there are two extremes. Whenever you see two extremes and at least towards periphery, my dear children, that means both the characters are being extreme characters are being selected. Okay, now this is known as disruptive method. This is known as disruptive method. Now over here, if the graph was something like this, that means the middle thing, whatever middle phenotype was there, that is being selected. It is known as stabilizing. Okay, now if suppose the graph is like this, that means only one phenotype is being selected. This is directional. So here disruptive, as in if you see, it is breaking down to two and the two extreme characters are being selected. So I'll say disruptive. Got it? And with that, we complete all the questions from evolution as well, and now we get ahead with human health and diseases, my dear children. So over here, what are the questions that we have? Let's see. First question, which of the following pairs contains an infectious and a non-infectious disease respectively? It should be infectious, One should be infectious and other one should be non-infectious. So typhoid and both are infectious, my dear children. AIDS is a sexually transmitted disease as well. Typhoid can be spread through oral fecal route, as in the food, water, everything. Pneumonia and malaria, again something that can be transferred. Cancer and malaria, cancer is not something that is infectious. So AIDS and cancer is in that category, where AIDS is infectious and cancer is non-infectious. Okay, going ahead. An intestinal parasite which causes blockage of the intestinal passage and whose eggs are excreted along with the feces of infected person is. So, intestinal parasite which is going to cause blockage in the intestine, there is one very important one, and that is Ascaris lumbricoides. That causes ascariasis, my dear children, and it is also known as roundworm. It is also known as roundworm. Okay, chalo. So, ask helminthes. Now, which of the following is affected by infection of Wuchereria bancrofti? Wuchereria bancrofti causes filariasis or elephantiasis. Why do you call it elephantiasis? Because the people who have this, they have legs or limbs like elephant size. Okay, because you know what, the lymphatic vessels which are there, they are getting affected. What is the function of lymphatic vessels? So in our body, the extra fluid which is being leaking out of our blood vessels are being taken by the lymphatic vessels. Now suppose it is not being taken by the lymphatic vessels, it will accumulate over there and cause inflammation. So exactly something like this is happening. Here the lymphatic vessels are getting affected, which is why there will be inflammation towards the lower limb region, my dear children. Okay, so it is an infection of Wuchereria bancrofti that affects the lymphatic vessels, which usually drains out extra fluid in our body. Now, which of the following pairs correctly matches a disease and pathogen causing it? So, ringworm is caused by trichophyton. Ringworm is a worm, right? Is it? No, it's a fungal disease. So, here it looks like a worm. There in the skin you will be feeling something like this. Okay, ringworm is a fungal disease and it is caused by trichophyton, epidermophyton, microsporum, all the fungus. Okay, pneumonia is caused by Haemophilus pneumonia. No, Haemophilus influenzae. Malaria is caused by Ascaris lumbricoides. No, Ascaris is caused by Ascaris lumbricoides. Malaria is caused by Plasmodium. What about Salmonella typhi? Salmonella typhi causes typhoid. So all these things wrong except A. Next, hemozoin is a precursor of hemoglobin. Precursor of hemoglobin as it helps in the formation of hemoglobin. Toxin released from Streptococcus infected cells. Toxin released from Plasmodium infected cells. Toxin released from Haemophilus influenzae cells. My dear children, it is toxin released from Plasmodium infected cells. So in the case of malaria, where Plasmodium is the guy with the major role, okay, what does it do is that it affects the RBC. It goes inside the RBC and it starts to divide. And basically it feeds on something that is known as hemoglobin, and it breaks down into heme and globin. But this heme is kind of very toxic for the Plasmodium itself, so it converts into something else, that is hemozoin, and it's kept like that in the cell. But when the cell bursts, what is happening? The hemozoin is being released. Now this kind of thing, this is kind of toxic for the normal human cell, which is why we get fever and chills during malaria. Whenever this RBC bursts and release a lot of hemozoin, that's when we get a problem. Okay, so there is a toxin released from Plasmodium infected cell. Okay, next question. The human immunodeficiency virus, HIV, primarily attacks and destroys. So who is the one that has a major role in our body for immunity? It's the lymphocytes. It's the lymphocytes who play a very important role in our body. I'm not saying others do not, but they have a very important role. In that, there is T helper lymphocyte. You know, they name only. They help other cells. So what do they do is that they call other cells or they do something by themselves also. So T helper cells are something that is acting as a medium link between lots of immunological reaction, my dear children. So this virus primarily comes and affects the T helper cells. So if T helper cell is not there, lot of the reactions would be messed around. You get it? So this becomes your answer. It is not TC cells. TC is T cytotoxic cells. There are different type of T cells. So those are not the one that are primarily attacked. They are attacked, but this is not the one primarily attacked. The one that is primarily attacked is TH cells. One very important thing. First time when HIV comes into our body, they are going to get inside macrophages. So if the question was like, which is the factory of HIV, it is macrophage. So they get inside the macrophage, they use the machinery of the macrophage and replicate the number. They increase the number and then they go attack the T helper lymphocytes. Okay, so since macrophage is not an option given over here, you don't even have to think. So macrophage, it usually said that HIV factory. And this is the one that is actually affected. Okay, Going ahead with the 7th question. Which of the following is an autoimmune disease? What do you mean by autoimmune? Wherein your body cells are not able to recognize what is mine and what is not. So they are going to kill their own cells. Okay, so these type of diseases are autoimmune diseases, my dear children. Example is rheumatoid arthritis, that is given in your NCERT. Cool. Chalo, next. Antibodies pass through placenta and present in the colostrum, which protects the newborn from certain diseases. Is that? So there are 2 antibodies which are being very important for baby and mother. That is not baby and mother, basically the baby. So through the placenta, IgG goes, and through the mother in the colostrum, that is the first milk, yellow milk, IgA goes. Okay, so I remember it like GAMED, gamed. These are the antibodies, GAMED. So G is the one that goes through placenta. That is the first thing that forms, right? Placenta. Then A is the one that goes through milk. So your answer is IgG and IgA. What do you mean by Ig? It is immunoglobulin. You know, antibodies are proteins, so immunoglobulins. Okay, next. The site where lymphocytes interact with antigens and proliferate to become effector cells are. Effector, as in they are going to fight with the antigens, as in foreign particles, and they are going to show their effect. So these sites are obviously secondary lymphoid organs. In the primary lymphoid organs, they are obviously produced, they get matured, and they also become antigen sensitive, but they do not interact with the antigens over there in the primary lymphoid organs. They are just made there. So basically thymus and bone marrow and thymus are the primary lymphoid organs, so I can exclude this option. What else I have? I have thymus and lymph nodes, I have Peyer's patch and tonsils, I have bone marrow and spleen. Here thymus is involved, so I have to ignore that. Bone marrow is there, so again I have to, because they are primary lymphoid organs. Only Peyer's patch and tonsils are left behind, my dear children, and this becomes the secondary lymphoid organs where this thing actually happens. So lymph node is also a secondary lymphoid organ, and spleen is also a secondary lymphoid organ along with Peyer's patch and tonsils. But since they are given in another option, we cannot take it. So Peyer's patch and tonsils is the correct answer for this question. Now, hepatitis B vaccine is produced from. Hepatitis B vaccine is produced from. Now, this is given in the NCERT below the vaccination page, my dear children. Lots of people will not find it. So, this vaccine was produced with the help of genetic recombination, and it was produced with the help of yeast. So, it was produced with the help of yeast. Please remember it as such. Okay, this vaccine was produced with the help of yeast. So, genetic recombination was done. Okay, not genetic recombination, I mean recombinant DNA technology was being used, and then the genes were put over here and the yeast was made to produce it. Okay, as in vaccine, what do? You take an inactivated version of something, right? So, in hepatitis B vaccine, whatever protein is being, in hepatitis B, whatever protein is there that causes, immunologic response, that particular thing was taken, put in the yeast, and its number was increased with the help of yeast. Okay, and now obviously this can be used as vaccine, because whoever gets this, they will be producing some amount of antibodies in their body, and that is the role of vaccine. Okay, now, which drug is used as a medicine to help patients cope with depression and insomnia? Depression and insomnia, my dear children. So, you have 2 drugs given in your NCERT for that. One is barbiturates and other one is amphetamines. Both of them treat depression and insomnia. Insomnia as in not able to sleep at that point of time, this is being taken up. That is amphetamines and barbiturates. Morphine is sedative and a painkiller. It makes you sleep, and also it is what? A pain killer. Cannabinoids, they are having effects on your cardiovascular system. Cocaine, again it has an effect on your CNS, central nervous system. Okay, so this becomes your answer. Along with it, barbiturates, do not forget it. Now, a chemical, so there are 12 questions, I wrote 11, oh my god, sorry. So, a chemical structure is given below, identify its plant source. What is this structure? So, I always think of it as an O. And what is the only drug with O, my dear children? It is opium. And the plant that produces them is opium poppy, and the other name of that same plant is Papaver somniferum. Papaver somniferum. So, this is your answer for the thing that produces opium. Okay. So, chemical structure given below is opium, my dear children, and that is produced from, not opium actually, but just for you to remember it. Okay, it's morphine I think, and basically it is Papaver somniferum which produces it. I said something wrong, that is okay. So, I am zoning out because after solving so many questions it happens. So, yes, in opium, my dear children, you have 2 categories. One is heroin and one is morphine. So I said it's not opium, it's morphine. So just understand, it's one type of opium which is present over here. Okay, structure of one type of opium, and it is produced from Papaver somniferum, and my voice is completely gone. Okay, there's one more question. Okay, charas and ganja are the drugs which affect what? Charas or ganja, my dear children, they are cannabinoids. They are what? Cannabinoids. They are produced from cannabis sativa, and they are going to affect the cardiovascular system, your heart and everything. So this is the answer for this question. Charas, ganja, marijuana, everything comes under this category. I think there is one more question. Yes. Smack is a drug obtained from the what? Smack. Have you heard of smack? Now smack is none other than, you're talking about heroin only. You're talking about heroin, which is opium, okay? Now it has other name, that is smack, and it is produced from the latex, okay? Latex of the Papaver somniferum plant. Okay, the white color latex that you get, from that you get it. That is known as smack. Now, coke or crack is used for that thing, cocaine. Remember that also. Smack is for heroin, coke or crack is for cocaine. And now, with that, we are ending these many chapters, and now you saw the speed with which we solved MCQ questions, right? And if you are also over here for KCET, okay, and you have stopped your preparation for that, then I would recommend you to join our series, that is Abhyas KCET test series, my dear children. So you can do it post your PU2 if you want, or if you want to join, you can join it now also, either way. But what do you get over here? You get confidence over here. Well, apart from that, there are some practical things also. That is, there will be 35 full syllabus as well as chapter wise test. Okay, there will be video solutions, and there will be obviously analysis of past 5-year question paper, and this is all for rupees 1499. So if you're someone who is interested, the link is given in the description box below. And one more thing, if you're someone who is not very much prepared for your 12th grade even now, so there is PU2 test series that you will be getting for rupees 1. So it is actually 1498, but you're adding just rupees 1 for getting that series as well. So practice and perform really well. So I was supposed to say prepare, practice and perform, but yes, you should be practicing and practicing. And we will get ahead with the next thing, but you can take a 5-minute break and come back, or probably I will be anyways joining the video and showing it to you. You can take a 5-minute break and come back. Okay? So okay children, let's get ahead with the next chapter, that is microbes in human welfare, where I have chosen 11 questions. Let's see those questions. First question, compare the statement 1 and 2. So statement 1 says mycorrhiza is the symbiotic association of algae and fungi. Now is this statement true or false? Well, it is straight false. Why? Because mycorrhiza is a symbiotic association, but it is not between algae and a fungi, it's between a plant and a fungi, my dear children. So I'll say this statement is false. Now the second statement says they absorb nitrogen from the soil and pass it to the plant. Who they? As in you're talking about the fungi. But the fungi that is there in the mycorrhiza, is it absorbing nitrogen? No, it is going to absorb phosphorus, my dear children. So this statement again becomes wrong statement. I can say both 1 and 2 are false. D. Okay, always remember it is phosphorus. Now going ahead with the next question. Alexander Fleming discovered penicillin while working on. So I'll tell one thing. He was working on some bacteria. So the option that has virus, we can cut it down. Now that's the bacteria, right? So this bacteria was normally growing. But once he didn't wash the petri dish, and what he observed was that the bacteria was there, but along with that there were certain fungus that was being formed over here, and this fungus didn't allow the growth of this particular bacteria, because probably they might be releasing certain antibiotic means it is obviously against the bacteria, it does not allow the growth of bacteria. So what was this fungus? This fungus name was Penicillium notatum, which was releasing the most famous antibiotic, that is penicillin. And what was the name of this particular bacteria? Is it Streptococcus? It is E. coli? Is it Staphylococci? What is the name of the bacteria? It is Staphylococci, my dear children. So this becomes the correct answer. Okay. Yes. Next. Which microorganism is useful in production of citric acid? Citric acid. So first thing, in our book, acetic acid is produced by this bacteria, and butyric acid is produced by this bacteria is given. So what is the name of the bacteria? Acetic acid is produced by Acetobacter aceti. Okay, butyric acid is being produced by Clostridium butylicum. Now you are asked about citric acid. Now this is an acid that is not produced by bacteria but a fungus. And who is the fungus over here? You might think that this is also fungus, this is also fungus. Okay, so Penicillium notatum is obviously a fungus, but it releases penicillin. Now over here, what are we left with? Aspergillus niger. So this is the one that produces citric acid, my dear little champions. Please remember, it's the fungus that produces citric acid. Going ahead. Activated sludge is the sediment deposited in. So you know what, whenever you want to treat the sewage, you make it undergo the first thing, that is primary treatment, where you have two things. One is filtration and other one is sedimentation. In filtration, you remove everything that is floating, and in sedimentation, you let the big things sediment down. So what are you left with? You are left with something that is known as primary effluent. Now, you take this primary effluent for secondary treatment. And what happens in secondary treatment? In secondary treatment, you have aeration tank, my dear children, where you put lots of air to basically break down whatever organic matter is there in this particular effluent. Now, since there is an aeration tank, obviously the aerobic bacteria will be growing up over here. And they will actually group up with some fungal filaments and everything to form a mesh-like structure that is known as flocks, which basically together act on the organic matter and try to break it down. But later, after everything is been done, okay, after the organic matter, everything is broken down, what happens is that we are going to put it in the next tank, that is a settling tank. And what happens in the settling tank? Whatever aerobic bacteria and the fungal filaments are there, the mesh-like structure, they will be sedimenting down. And whatever, let's say, all corrected fluid which is there, the perfect fluid which is there, that is left behind. Now that is being put down to river, sea, whatever you want to do, because that's all cleansed. Now whatever is left behind, you will call this as activated sludge, my dear children. You will call this as activated sludge. Okay, now some part of this activated sludge could be put down back into this aeration tank for the next set of primary effluent that is going to come. Okay, because you need some aerobic bacteria, right? So you get it from here. Now, whatever is left, you take it to an aerobic sludge digester later on. So this question is, activated sludge is the sediment deposited in. So you are asked about this tank. What is this tank? This tank is settling tank, my dear children. Later, this activated sludge is passed down to anaerobic sludge digester, where you would be having anaerobic bacteria that will be acting on the aerobic bacteria and producing methane or H2S and carbon dioxide and all these biogases. So this is sediment deposited in settling tank. Please remember it. Going ahead, match the column 1 with column 2 and select the correct option using the codes given below. So citric acid we just studied now, it is produced by Aspergillus niger. So A goes with 3. So A goes with 3, there are 3 options, we can cut this option. Now cyclosporin and butyric acid also I told. So butyric acid is produced by Clostridium butylicum. Okay, so D goes with 2. So here there is only 2 options, but D goes with 2. I can again cut this option as well. So you have to choose between the 2 options that you have. Now cyclosporin A and statins is there. Cyclosporin A, my dear children, it is again produced by a fungus that is Trichoderma polysporum. Okay, Trichoderma polysporum. It is an immunosuppressant, as in it suppresses our immune system. Okay, for example, we have an organ transplant. At this point of time, we do not want the organ to be damaged by any sense. Our body will feel that it's foreign, I have to kill it. But it should not do that. At that point, we take immunosuppressant. Cyclosporin A is an immunosuppressive agent and it is produced by Trichoderma. Now, talking about statins. Statins is the one that stabilizes our blood cholesterol and it is produced by Monascus purpureus, that is C goes with 4. The C1 will be going with 4. So, I have an option like this, where all the things match and this becomes your answer. So, in 5th question, B is your answer. Okay, this is how you would be able to remember the functions and everything, otherwise there is less chance. Okay, now during sewage treatment, the gas produced in anaerobic sludge digester includes, I told you, what do? You take the activated sludge and then you put it in anaerobic sludge digester, where the anaerobic bacteria makes it produce biogas. Now you're producing biogas. So what does this particular biogas include? The major constituent of the biogas would always be methane. Apart from that, there will be hydrogen sulfide, H2S, which gives a foul odor, and also a little amount of carbon dioxide as well. Now, this is a constituent of biogas. So, whenever you talk about biogas, it means you're talking about majorly these 3 gases up there. Okay? Now, next, which of the following is not an advantage provided by mycorrhiza to the plants? Not an advantage. Okay? So, resistance to root-borne pathogens. Do you think mycorrhiza, do you think it provides resistance to root-borne pathogens? Yes, it does. Okay. does not allow pathogens to grow in there. Now, does it increase in yield quality? No, my dear children. It has no effect on the yield quality, that is whatever harvest would be coming, okay, that is not managed by mycorrhiza. Now over here, tolerance to salinity and drought, yes, that is there. Overall increase in the plant growth and development, that is also there, but this is not something managed by mycorrhiza or provided by mycorrhiza to the plants. Now this is not something that I made up, this is given in your NCERT. So yield has nothing to do with mycorrhiza, please remember that, okay. Yes. Next, the microbial biocontrol agent for butterfly caterpillar is, what do you mean by biocontrol agent? So basically, you know, pesticides, we put pesticides on plants to kill the pest, but we don't want to, because these chemicals that we are putting will be going into the soil, from the soil it will go into the water, and this is the water we drink, so we are taking the chemical. So instead of that, why not use biocontrol agents, as in organisms that would be eating this pest, that is the best thing, no, or probably organism that could affect these pests which is there. So the microbial biocontrol agent for butterfly caterpillar is, so there is a microbial agent that would be killing the butterfly caterpillar. Do you know what is it? Is it Bacillus thuringiensis, Saccharomyces cerevisiae, or basically cyanobacteria, Staphylococcus? What is the answer for this? This is the very famous Bacillus thuringiensis, which you also call as BT, my dear children. What does BT do? It has a gene which is known as cry gene, and cry gene, my dear children, produces a toxin substance, okay, in the bacteria. But in the bacteria, this is not in the active state, it is in inactive state, it is in protoxin state. Now where does it go become active? Basically, I am a caterpillar, I come and eat the plant which is having this bacteria. What happens? I am taking this bacteria. Now when this bacteria reaches to my gut, what will happen? The toxin which is there, it will turn to active toxin. The protoxin turns into active toxin. Why? Because I am an insect, and in my gut there is alkaline pH, and in alkaline pH, my dear children, what happens? The particular toxin gets active. So it is Bacillus thuringiensis which is responsible for killing the butterfly caterpillar, okay. Now, or basically the insects, okay. Now, which of the following statements regarding phlox is incorrect? What do you mean by phlox? Remember, it's a mesh-like structure found by aerobic bacteria, as well as certain fungal filaments, as well as certain other extracellular things which are there. So you have to find what is incorrect. They are the masses of bacteria associated with fungal filaments. Is it true or false? Yes, it is true. Now, they are produced in aeration tank, as in where the air is there. Obviously, that's why aerobic bacteria is formed. Now, they consume major part of organic matter. Yes, they are going to attack the organic matter and break it down. That is also true. Now, they are found by useful anaerobic microbes. Anaerobic microbes are, no, they are produced by, sorry, they are made by aerobic bacteria, no. So how can you say anaerobic microbes? So this is a wrong statement, and this is the incorrect one that we were looking forward for. Okay, cool. Children, are you understanding? I hope you are. Now, select the correct statement from the following. Biogas is produced by the activity of aerobic bacteria on animal waste. Please, my dear children, please remember, biogas is not produced by aerobic ones. It is always produced by anaerobic. Okay, when there is no oxygen, that's when the production of biogas increases. Okay, so this itself is wrong. You have to find what correct statement. So this is a wrong statement. Methanobacterium is an aerobic bacterium found in the rumen of cattle. Now methanobacterium is the one that produces methane, and methane is a part of what? Biogas. So can it be aerobic bacterium? No, it will be anaerobic bacterium that would be found in the rumen of the cattle. Okay, that is actually found in the rumen of the cattle, but it is anaerobic. So this again is wrong. Now biogas, commonly called gobar gas, is pure methane. Is it pure methane, my dear children? No, it is H2S and also carbon dioxide. So this again is wrong. Now activated sludge in the settlement tanks of STPs, that is sewage treatment plants, is a rich source of aerobic bacteria. Yes, activated sludge contains aerobic bacteria, fungal filaments and everything. So it is a rich source of aerobic bacteria. So I'll say this is the only statement that is correct. Okay, you know you have to find the correct one. No, I don't know what I was doing. So basically this is the correct one. Okay, I put double tick here. I'll put wrong like this so that you don't. Okay, cool. Now, bioactive molecule cyclosporin A is produced by, I told you cyclosporin A, remember trichoderma. Trichoderma is a fungus, my dear children. Its name is Trichoderma polysporum. I'll tell you one more thing about trichoderma. That is, Trichoderma is a fungus, and this fungus stays in the roots of the plant, and they do not allow the growth of another fungus. So it is also acting like a biocontrol agent. It also acts like a biocontrol agent. Please remember that. And with that, we complete all the 11 questions. Please remember, wherever you have certain things, for example, products, okay? For example, yeast produces what? Yeast is going to help in the production of bread and also wine, that is brewery and things like that. Now there can be a question that which among them are produced with the help of distillation. So brandy, whiskey, beer, and then wine is given. You are asked which one is produced through distillation. Distillation means basically you're taking the water content, whatever is there, it is going in the form of water vapor. You're getting concentrated form of alcohol. That is the process of distillation. So which of them you are not getting through distillation? This beer and wine. That is why the amount of alcohol in them would be comparatively lesser than brandy and whiskey. Now you could be asked about Roquefort cheese. Okay, what is that organism that is responsible for production of Roquefort cheese? It is a fungus. Okay, now Swiss cheese is asked. In Swiss cheese, my dear children, who is responsible for production of Swiss cheese? It is a bacterium, that is Propionibacterium shermani. Okay, so please remember all this thing, and it releases lots of CO2, which is why there are big holes in the Swiss cheese. Now in the milk, what is milk to curd formation? What bacteria is there? Lactic acid bacteria is there. And what quality does it add to the curd? It helps in the production of vitamin B12 in the curd. So all the small things are there, which is very much important, and you should know it. Okay. Statin is produced by Microsporum Monascus purpureus. Remember that also. Chalo. Next chapter, that is biotechnology, principles and processes, my dear children. 11 questions again. Are you ready to solve it? So, my dear champions, I hope you are in a mood to solve a little more MCQs, and then we are done with MCQs. Okay. So which of the following is not a feature of plasmid? Which of the following is not a feature of plasmid? You know what is plasmid? So suppose this is an E. coli. E. coli will have its own circular DNA that is there, but apart from that, it also has its extra chromosomal DNA, as in something apart from it that is extra chromosomal, and it will be also circular, and it will be double stranded, and it is going to undergo replication as well. It will have its own replication apart from this. So it is extra chromosomal, it is circular, but it is not single stranded, it is double stranded, my dear children. So this is not a feature of plasmid, and it shows independent replication. So this is your answer. C is your answer. Okay. Now next question, which is there. The procedure through which a piece of DNA is introduced into a host bacterium is known as. So you are having a DNA, vector DNA, and you're going to put it in the host bacterium. What do you call this process as? Amplification? No. Cloning? No. Transformation? Yes. Restriction? No. So you're transforming that particular organism, host organism, right, with your piece of DNA. So that is transformation. Cool. Now next question. The commonly used vector for cloning genes in animals is, so if you want to put something in animals like us, what is the vector that you will choose? Will you use rhinoviruses? No, my dear children, I won't use rhinoviruses. I will use disarmed retroviruses. So first thing, for basically putting something into animals like human beings or things like that, we definitely need a virus only. Okay, because virus has a specialized mechanism. So we cannot take something that is armed, as in something that has its own potential to cause some trouble to us. We will take a disarmed retrovirus, as in something that would not be causing trouble to us. So disarmed retrovirus is taken, okay, for basically acting as a vector for cloning genes in animals. Now what about disarmed TI plasmid? No, TI plasmid is used usually in the case of plants, my dear children, and Agrobacterium tumefaciens has this TI plasmid. So again, these options are not eligible for this particular question. Okay, always remember it's a virus but not rhinoviruses. Rhinoviruses, remember, they are the ones that causes cold. So it's disarmed retroviruses that are responsible for acting as a vector. Now restriction site of EcoR1 enzyme is, it's a very famous question. So restriction site is 5-GAA-TTC3. So sometimes we forget 5 and 3. Always remember all the processes in replication, whatever is goes from 5' to 3'. Similarly here also EcoR1's restriction site is 5-GAA-TTC. Cool. Next. The extraction of separated bands of DNA by cutting out them from agarose gel. So, in gel electrophoresis, you make the DNA run through the chamber. Right? Now, you want to extract this DNA out. What do you call this process as? You will be calling this process as elution. Okay, what will you call it? You will call this elution. That is the process of extraction of DNA bands. Okay, by cutting them out from the agarose gel. Now, next thing, the thermostable enzyme DNA polymerase is obtained from, thermostable as in it will be able to survive at higher temperature. So, this type of DNA polymerase would be obtained from which organism? Agrobacterium tumefaciens, Thermus aquaticus, Escherichia coli, Meleidigine incognitia. So, it is the very famous Thermus aquaticus, which is why the name of the DNA polymerase which is extracted from them, it is known as TAC polymerase, as in Thermus aquaticus polymerase. It will be able to withstand higher temperature, and in the process of PCR, that is polymerase chain reaction, you need a DNA polymerase that can go up to higher temperatures. Okay, because the whole process happens in 90, 92 degrees Celsius and 52, 55 and 72, 70. So, it's high temperature, you get it? So, this becomes your answer. Next question. Agrobacterium tumefaciens transform normal cells into a tumor by delivering a piece of DNA known as. So, Agrobacterium tumefaciens, my dear children, it is a bacteria, and that is going to transform a normal cell into a cancerous cell or tumorous cell. How? Basically, this Agrobacterium tumefaciens has a plasmid. This plasmid's name is Ti plasmid. What do you call it? Ti plasmid, my dear children. Now, this Ti plasmid has a segment of DNA which is known as transfer DNA. This piece of DNA is known as transfer DNA, my dear children. And what does this Agrobacterium tumefaciens do? When one particular organism gets this particular plasmid having the transfer DNA, okay, this transfer DNA will go and affect the whole cell cycle and things like that, and it will be causing tumor. So, Agrobacterium tumefaciens transform normal cells into a tumor by delivering a piece of DNA which is known as tDNA or transfer DNA. Now, what can I do? If I want it to be used as a vector, what do? I remove the transfer DNA and I am going to put my gene of interest over here. Anyways, this whole thing can be transferred into a plant now. You get it? So, that is why Agrobacterium tumefaciens is acting as a vector for plants. Okay, disarmed ones. Now, going ahead, identify the correct statement. HindII cut the DNA specific sequence of 6 base pairs. Yes, HindII was the first restriction enzyme to be found, and it cuts at a sequence which is having 6 base pairs, my dear children, just like EcoR1. Now, next, microinjection is a technique of introduction of rDNA into the plant cell. Microinjection as in you are going to insert the DNA with the help of an injection directly into the nucleus of the organism. So, is it done in the case of plant cell? Is that how you introduce recombinant DNA into the plant cell? No, my dear children. Okay, this is usually used in the case of animal cells. In the case of animal cells, not in the case of plant cells. Now, biolistics is a technique for the introduction of rDNA into animal cell. What do you know biolistic? Where you are using the gene gun, my dear children. Now, where do you think you would need a gene gun? Where the cell wall is there. For penetrating into the cell wall, it's difficult, which is why you need a gene gun that would transfer the genetic material inside. So, bialistic is not a technique for animal cell, but it is for plant cell. So, they have interchange. So, both of these statements are wrong. Over here, this is the correct statement, but let's read this also. Retroviruses are commonly used cloning vectors in plants. I already told you it's not for plants, it's for animals. So, this again is wrong. This is the only correct statement that we are having. I don't know why is it marked like that, but the answer is this. Okay. Now, which of the following is a stain used to visualize DNA fragments in gel electrophoresis? So, when the DNA runs through gel electrophoresis, you cannot obviously see it. So, for viewing it, you need something. You need a stain, and that stain is what? Cotton blue, ethidium bromide, acetocarmine and safranin, which is that it is ethidium bromide, my dear children, and you cannot, even after staining, you would not be able to see it unless and until you are going to put it under UV rays. So, under UV rays, this particular ethidium bromide would be appearing some like bright orange color, okay? It would be appearing like some bright orange color. Please remember etherium bromide. Okay, so Br and O, bro. Okay, bright orange. Works? Chalo. Next, in the given diagram of PBr 322, which is a plasmid, my dear children, vector, identify the restriction sites A, B, C and D. So over here A, B, C and D is being mentioned over here. So we have to find what is it exactly. So I'll tell you one thing. The most smallest version of thing will be obviously this would be a ORI site, origin of replication site. Now near the origin of replication is very much needed for the DNA polymerase to come and attach. Similarly, you need a protein also which is known as ROP protein. So this is ROP protein site and this is origin of replication site, my dear children. Now if you go ahead, this site, the C site which is there on towards this is always tetracycline resistance, for tetracycline resistance. Okay, now over here if you see this B site which would be nearer to the origin of replication, this would be ampicillin resistant site. Okay, that means this particular plasmid is resistant to ampicillin and also resistant to tetracycline. Both of them are antibiotics. Now if you see D, now D is a site where there are two things like this. Now this is a site for HIND3, this is a site for HIND3, my dear children. Okay. So you should be knowing the structure as a whole. So please do not forget to go through the whole structure of PBr 322. Okay. So this is your answer. So according to this, A is ORI. So there is only one option with A as ORI and that is this. If you know A is ORI, you would be able to answer the whole thing. So B is now what? Ampicillin resistant gene. And now C is what? It is tetracycline resistant. And D will be what? D. Where is D? Yeah, D will be HIND3. Cool. So this becomes your answer. Fine, going ahead. Statement 1 says plasmid DNA acts as vector to transfer the piece of DNA attached to it. So plasmid DNA which is there, my dear children, it will be acting as a vector, as in something that would be helping in transportation of some other thing. So basically plasmid DNA acts as a vector to transfer what? The piece of DNA attached to it. Suppose I have put some DNA over here. So obviously it is acting as a vector. So I will say this is correct. But what about the statement? Vector DNA act as plasmids to transfer the piece of DNA attached to it. Is it? Vector DNA, how can it act as a plasmid? Vector DNA is going to insert its content into the plasmid, right? Okay. So how can, just a second, okay, just a second. I think there is a small confusion here. Yes, so we were here. Vector DNA acts as plasmids to transfer the piece of DNA attached to it. No, it would not be true. Okay, so this would be a false statement, even though it's kind of confusing. Okay, if I find something, it's kind of confusing because it has some, correctness to it, but I'll research to it and I'll let you know if at all there is a correction. I always do that. Okay, so I'll let you know, but as of now, I feel it's a very wrong statement. So, vector DNA is not going to act like plasmid to transfer the piece of DNA attached to it. This is the piece of DNA, vector DNA what is there, that is the piece of DNA that is attached, right? So how can it transfer something attached to it? So obviously I'll say statement 1 is true, statement 2 is what? False. So over here if I click, something will come or not? No. So I have to move towards that. Yes. Okay, I hope you saw the answer. It is statement 1 is true and statement 2 is false. Yes. Now, next chapter, that is biotechnology and its application, where I have taken 10 questions, my dear children. Let's see those 10 questions. First question. In human insulin, the 2 polypeptide chains are linked together by. The 2 polypeptide chains, my dear children, how will that be linked together? With the help of covalent bonds, with the help of glycosidic bonds, phosphodiester bond, disulfide bonds. The answer is disulfide bonds. Okay, 2 polypeptides are linked together with the help of disulfide bonds. Again, a straight question, no need of thinking much. You have to remember it, that is a different thing. Now going ahead, ADA deficiency in humans could be permanently cured by, what do you mean by ADA? ADA is an enzyme, adenosine deaminase. Okay, now this enzyme deficiency can be cured by what? Bone marrow transplantation. If I transplant the bone marrow, will it work? Introduction of functional ADA through injection. No, that's not any good reason to do that. Okay, basically it's not a good way. Now, introduction of gene isolated from marrow cells producing ADA into embryonic cells. So, I am taking the gene from the bone marrow cells, and I am going to put this gene into the embryonic cells, my dear children. Now embryonic cells has the capacity, whenever they divide, they are going to give this gene to the next cell as well. So all the cells of our body would be having it. So then it is fine. Obviously that would be a permanent cure. But how do you know it? Only if you do the testing, that's when you would be knowing it. In the embryonic stage itself, you have to do the testing. That's how you can do it. Otherwise there is no way. And that's kind of very difficult, right? So enzyme replacement therapy also would not be working. This only cure, permanent cure is C. Okay, you have to take the gene and you have to put it into the embryonic cells. Now, going ahead, which of the following statement is correct with respect to synthesis of insulin? So, it is synthesized as proenzyme and needs to be processed to become functional enzyme. First thing, is insulin an enzyme or a hormone? It is a hormone, my dear children, which is produced by pancreas. So, it is not synthesized as a proenzyme. So, wherever enzyme term is written, I can just say it's wrong. So, these two statements are wrong. Now, synthesized as prohormone and needs to be processed to become functional hormone. That would be correct, but let's read this also. Synthesized as prohormone and needs to be processed to become functional proenzyme. That is wrong. But suppose this statement was put over here and this was put over here. When you read it in haste, you will feel that first half is correct, so I will give correct answer. This would be the correct answer. But no, you have to read the entire option when the options sound so similar. Okay, so this is your correct answer. So insulin, my dear children, comes in a pro version which you call as proinsulin, and then you process it to become insulin, the functional one. Okay. Now next one, the silencing of mRNA has been used in producing transgenic plants resistant to what? First thing, why do you need to silence the mRNA? You know, mRNA, it undergoes translation to form proteins. Now suppose I do not want these proteins to be made because these might be toxin substances. So at this point of time, what can be done? I can silence this mRNA by introducing another strand of RNA. So when the RNA is in double strand, obviously this cannot undergo the process of translation. So that is what you mean by silencing of mRNA, for which what do you use? You actually use a double stranded RNA. In this double stranded RNA, one strand will be coming and attaching over here, and the other strand will slowly degenerate. So silencing of mRNA has been used in producing transgenic plants resistant to what? White rust, bacterial blights, bollworms, nematodes. The answer is nematode, because you know there is a nematode, that is Meloidegain incognitia, that basically affects the tobacco plant. Okay, that produces some certain toxic substance in the tobacco plants, basically not allowing it to grow very well. So in that plants, if you are basically giving a DSR, double stranded RNA, obviously what will happen whenever the nematode will be introducing its own mRNA, okay, what will it do? Basically, this from double stranded RNA, one RNA will come and make double strand over here and would not allow the formation of proteins up there, and hence it would not be able to affect. Okay. So I'll just repeat once more, listen to me carefully. This nematode which is going to come and attack the tobacco plant, right? So when it eats the tobacco plant, when it is eating it, it is getting this double stranded RNA. Now from this double stranded RNA, one RNA would be going and attaching to its RNA, mRNA, and forming a double strand there. Now will it be able to produce protein? No, it won't be able to undergo the process of translation. So silencing of mRNA has been used to produce transgenic plants resistant to nematodes, my dear children. Okay, works. Chalo. Now, corn borers are controlled by protein encoded by the genes. Corn borers. Now, these are insects. Now, insects are being controlled by a gene, that is cry gene, and cry gene belongs to Bacillus thuringiensis, if you remember. Okay, it belongs to Bacillus thuringiensis. But which form of cry gene? You have so many cry genes over here. So, the answer for this is cry1AB. Okay, cry1AB, which is mentioned over here, is the one that is going to produce toxins that could kill corn borers. Now, what about if the question was cotton bollworms? Then it would have been cry1AC and cry2AB. So 1AC, how do I remember this for cotton bollworm? So first letter is C, that is cotton, and the second letter is B, that is bollworm. Now whole sentence, if you see, this is very long. Here also this would be very long than corn borers. So this is for cry1AC, cry2AB. Now over here this is also C and B. So as I said, this is long. Cotton bollworm, the name is also long. Over here C corn borers, sorry corn, and over here borers. And there is only one thing that is there that you have to remember, gene. I know it's complicated, but yes, it's important. Okay. Yes, next, Meloidica incognitia is what? I just told you it affects the roots of the tobacco plant. So it is a roundworm, I will call it a nematode instead. Okay, so Meloidica incognitia is a protozoa, nematode, or a fungi, or an algae. It is a nematode. So roundworms comes under nematode category, but I would be better in a better manner if we talk about it, then we'll just call it a nematode instead. Okay, so it is a nematode and that affects the tobacco plant. Going ahead, crystalline protein synthesized by Bacillus thuringiensis is activated by. So this toxin, which is inactive, it goes and gets active in the gut of the insect. I told you, but what gut? Mid gut or foregut? Is it acidic pH or alkaline pH? It is alkaline pH, my dear children, and it is also mid gut where it gets activated. So your answer is C. Small details would be missing in our answers, so which is why we put MCQs instead. So in MCQs you get a lot of information and you can put it directly into the answer also whenever and 1, 2 marker, 3 marker comes in. Okay. Now insect tolerant gene from Bacillus thuringiensis is introduced using TI plasmid of. Now I am taking this Bacillus thuringiensis gene and I am going to put it inside some plant. For that I need a vector. I need what? A vector, my dear children. So now this particular vector could be what? TI plasmid, obviously. Now TI plasmid belongs to what? Which are the organisms which is mentioned over here? Agrobacterium tumefaciens, Escherichia coli, Meloidica incognitia, or Salmonella typhi. The answer is Agrobacterium tumefaciens. Okay, the answer is Agrobacterium tumefaciens. Insect tolerant genes for Bacillus thuringiensis introduced using TI plasmid, that is of Agrobacterium tumefaciens, where you would be removing the T DNA and you are going to insert the insect tolerant gene of Bacillus thuringiensis. Cool? Yes. Now in the tissue culture, to get virus free plants from diseased one, which part has to be chosen? In tissue culture, to get virus free plants from diseased one, what part would we choose? Always remember, my dear children, in tissue culture, whenever you are doing this and you want virus free plant, you would be taking 2 meristem. One is apical meristem and the other one is axillary meristem. You can take either of them. So over here only apical meristem is there, so I will just take this. You know, apical meristem is the one that is present in the shoot tip and the root tip, that helps in the shoot growth as well as the root growth. Okay, so to get the virus free plants, I have to choose the apical meristem, which will be grown under tissue culture. Okay, next, the therapy given to cure adenosine deaminase deficiency comes under the category of chemotherapy, gene therapy, radiation therapy, immunotherapy. So in them, what is happening is that the gene which will be coding for adenosine deaminase, that has been put inside the lymphocytes. Okay, in the lymphocytes that has taken from, for example, in my body there is no adenosine deaminase gene. My lymphocytes are taken and then this gene is being put up and then these lymphocytes are put up back into my body. Now this type of therapy where I am inserting the gene and doing some alteration over there, you will call this as gene therapy. Again, a very easy question, but it comes and you might get confused if you do not know it at all. Okay, it will be gene therapy. Going ahead. With organism and population, having 7 questions, but I just want to go back to the question which is there. Just want to have the number of that question because I don't want to teach you anything that is wrong. Where is that statement? Yes, 11th question. Okay, fine. Going ahead with organism and populations. Let's start. So there are 7 questions up here. Now the shape of the age pyramid reflects the population growth status. In this context, the given age pyramid indicates that the population is what? This is pre-reproductive, this is reproductive, and this is post-reproductive. So over here if you see, pre-reproductive number is really less. That means probably there are very few people who would be reproducing in the future. So population rate is obviously declining. It is not increasing, it is not stable, it is going to decline because this number is less. So this would be declining. Cool. Now Jiva was growing a bacterial colony in a culture flask under ideal laboratory condition where the resources sooner or later became limiting. So over here one thing is clear, that the resources are limiting, it's not exponential growth. So in exponential growth we believe that there is nothing to stop, so the growth goes on like this. Now in logistic growth, my dear children, what happens? The growth initially takes some time to grow, and then it grows and then it reaches a limit. Now this limit is known as carrying capacity of the population. Okay, this is the carrying capacity because there are certain limited resources. That means there is no much food, there is no much air, there is no much water and everything to sustain everybody, which is why we consider this to be ideal. So over here it is mentioned the resources sooner or later became limiting, my dear children. That means you're talking about logistic growth, not exponential growth. Now which of the following equations will represent the correct growth in this case? So this is an equation for exponential growth, my dear children. Now if you're looking for logistic growth, the equation is this, that is dn by dt is equals to Rn by k minus n by k, where k is the carrying capacity, my dear champions, r is the rate of intrinsic increase and n is the population size. This is the size after time, okay, after some time. So this would be the correct answer. Okay. Now, the adaptation in which an organism matches its color with the surrounding to get protection from predators is known as. So, for example, I am over here, okay, I am over here, from this point onwards nothing is being seen because I am camouflaging. Okay, me and the board is basically camouflaging. So, this is an example of camouflage, okay, and an organism uses it to safeguard themselves. So, you might have seen chameleon changes its color so that no one can easily spot it, okay. So, this is known as camouflage. Okay. Next question. Monarch butterfly is avoided by the predators because, there is a butterfly which feeds on something really distasteful during its larval stage, which is why it also turns out to be distasteful. Okay, it is very distasteful. So anyone who comes and eats is like, I don't like it. I won't be coming and eating it. So in that way it is avoiding predators, right? Obviously. So it is cryptically colored? No, it is highly distasteful, my dear children. This is the answer. It produces a poisonous cardiac glycoside? No. It has stinging cells on its body? No. The answer is it is highly distasteful. Okay? Fine. Next. On the rocky sea coast of Scotland, the larger and competitively superior barnacle, that is Balanus, dominates the intertidal region. It dominates the intertidal region, my dear children, okay, in midden, and excludes the smaller barnacle, that is Thalamus, from that zone. So it is saying that, hey Churku, whoever you are there, just move around, just move out of this area, this is my area. So what kind of interaction is being shown over here? Is it predation? In predation you basically kill the prey, right? Is it parasitism? No. Is it commensalism? Is it helpful in any ways? No. It is competition? Yes. Over here, the first thing, competitively superior barnacle, that means they are doing competition, my dear children. So your answer becomes D. Okay, they are doing competition, which is why they are making, you have this term that is known as competitive exclusion. You are making the smaller ones move to different area because they want to occupy the larger area. You get it? Yes. Now, in a population with a large number of older, in a population with large number of older individuals than younger ones, it can be predicted too. Now there are large number of older individuals, as in the ones who might be in the, in the reproductive or post-reproductive stage rather than pre-reproductive. So obviously if pre-reproductive is lesser, I will say this is declining. Again the same graph that was shown over here. Okay, the same thing is there in different language. There they gave the picture, here they didn't give the picture. Okay, so if younger ones are less, that means your population is declining because they won't be reproducing, there is no one to reproduce. Now again a question related to that. What is the shape of the given age pyramids reflect about the growth status of the population? Okay, so let's see. Over here, in A you see the pre-reproductive, it's very much. And over here the pre-reproductive is kind of same, equal to the reproductive, and here it is very less. So I will say this is the one where the population is growing because pre-reproductive organisms are more in number. So they would be able to reproduce and give rise to more organisms. So I would say this is what an expanding thing. So this is expanding. Okay, wherever this is stable, this is what? stable. What about this? Since pre-reproductive organisms are less, that means there won't be much amount of reproduction happening. That means this population would be declining. So this becomes your answer. Okay? And with that, we are covering organism and populations as well. Now we are going ahead with the next chapter, that is ecosystem, my dear children. Again, you have 10 questions to solve. Let's solve all of them. So rate of biomass production is called, the rate of biomass production, you call this as productivity. Okay, now when this is done by producers, when the producers produce, you call it primary productivity. Now when the consumers, the first consumers produce, you call it secondary productivity and things like that. Okay, so rate of biomass production is known as productivity, a very, very easy question. Going ahead with the next question, GPP minus R is, what do you mean by GPP? That is gross primary productivity, as in the plants, they would be producing some organic matter, that is known as GPP, gross primary productivity, but they would be utilizing some of it, and that is known as respiratory losses. That is known as respiratory losses. Now what is left behind? Okay, what is left for the next organism? That is net primary productivity. So after utilizing whatever is left for the next trophic level, my dear children, that will be net primary productivity. So GPP minus R is net primary productivity. Okay. Now the rate of formation of new organic matter by producers is, so new organic matter, my dear children, and producers are doing it. So what do you call this? This is gross primary productivity. Now when you minus it with respiration losses, you get net primary productivity. So here that is asked what rate of formation of new organic matter. So this will be the answer. Where is it? Yes. Okay. Going ahead to the next question. Identify the correct sequence of steps in the process of decomposition. Now in decomposition, first thing, you know, detritivores which are there, they will be eating up the food and they will be breaking down into small fragments. Okay, this is known as fragmentation. Now during this time, whatever water soluble inorganic things are there, they will be leaching down into the soil, my dear children. That is known as leaching. After that, my hair, okay. After that, the organic matter which is still present in the smaller units, my dear children, have to be broken down to inorganic matter. And what do you call this process as? You call this process as catabolism. You're cutting them down. Okay, this is organic to inorganic. You're not breaking big things into small things. That is fragmentation. Organic to inorganic, when you're breaking it down, it is known as catabolism, that is done by decomposers. Now, during this process, it's not a one-day process. It does not happen so fast. So, basically, they take a pit stop and they form something that is known as humus. So, humus is like, it is like partially broken down version of the organic compound. So it will be rich in organic compound but not way too much, but yes, it will be there. Now this humus formation is known as humification, and after that slowly it will break down and will be giving down the minerals, that is known as mineralization. The correct step will be fragmentation, then leaching, then catabolism, then humification, and then mineralization. So the answer is A. Okay. Now next, identify the decomposers among the following in the pond ecosystem. A is bacteria, B is fungi, C is flagellates, and D is phytoplankton. So decomposers, my dear children, bacteria and fungi, or you say FB, Facebook, I remember like that. Those are the famous decomposers, even though others can also, flagellates can also act like decomposers, but no, I'll mostly associate with something else, but not decomposers. So I'll say the only one is A and B. A and B only would be the correct, most correct answer for this. Phytoplanktons are producers. Okay. If the energy produced at the level of producers is 1000, the energy available for the secondary consumer is. So producer, my dear children, is eaten up by the primary consumer, and the primary consumer is being eaten by the secondary consumer. So if you are said that producers has 1000 joules of energy, how much does the secondary consumer have? That is the thing. So you know, in energy transfer, we apply 10% law. So 10% of the energy would be going to the next trophic level. So 1000's 10% would be what? 100 joules. And 100 joules 10% would be what? 10 joules. Because rest of the energy would be used by themselves and also would be lost when the energy transfer is happening. So you only pass 10% of the energy. So I'll say the secondary consumer, you will be having just 10 joules of energy. Okay? Next. The process of breaking down of complex organic matter into inorganic substances. I told you it's not like bigger substance into smaller substances. It is complex organic matter into inorganic substances. You call this as fragmentation or what? No, you will be calling this as catabolism. B is your correct answer. Cool? Next, now the rate of decomposition is slower if detritus is rich in. So the dead and decaying matter which is there, it will take a lot of time to decompose if it is rich in what? Chitin, nitrogen, water soluble sugars, water insoluble sugars. So the answer is chitin, and chitin is mostly there in the cell wall of fungus and things like that. Okay. So chitin, if it is there, it is hard substance, it will be taking too much time to break down. Cellulose, chitin, whatever is there would be difficult. Okay, so even if cellulose was there, which is the cell wall component of the plant cell, still it will take so much time. Now next thing, each trophic level has a certain mass of living material at a particular time, and it is known as, for example, all the producers present at a particular time would be having a mass, right? What do you call this mass as? You will be calling this as standing crop, my dear children. This is known as standing crop. At each trophic level, certain mass of living material at a particular time is known as standing crop. Standing state is the amount of nutrient available at a particular time. Okay, so standing crop is the answer for this particular question. This is given in your NCERT. Now, next. Statement 1 says the pyramid of biomass in sea is generally inverted. Is it so? In sea, my dear children, who are the producers? Phytoplanktons are the producers. So obviously their biomass is very, very less. Now if you talk about zooplanktons, they would be having higher biomass. If you talk about fishes who eat them, they would be having more biomass. So obviously the pyramid would be, biomass pyramid will be inverted. It won't be upright, but it will be inverted like this. That is totally correct. The statement 2 says the biomass of fishes far exceeds that of phytoplankton. Yes, the fish's biomass would be more than obviously the phytoplankton. So both the statements, if I see, is correct only. So both statement 1 and 2 are correct. Now this is in the case of C, my dear children, please remember that. The pyramid of biomass is not always inverted, but it is inverted in the case of C. Chalo. And with that, we cover ecosystem chapter as well, and now it's time for the last chapter, which is biodiversity and conservation. Again, 10 questions. So how is the Josh? Are you enjoying? We are just left with 10 questions, my dear children, but I hope you are able to revise a lot of your concepts with the help of these MCQs. So you do have to let me know if I helped you in this case. And as I said, there's one question that I'm doubtful. The 11th question in that chapter, we will be sorting it out. Okay. So because my brain cells are about to die as of now, I am not able to even understand the question properly. So I have to go through it once more and I will tell you, okay. So as per species inventories, 70% of species recorded are. So 70% of the species which are there on earth, they are animals, my dear children. Okay, rest 22% or something like that is plants, but 70% most of the species are animals. Cool? Straight questions are there, you have to remember it. Now, the most species-rich taxonomic group among invertebrates, as in the one that does not have vertebral column. So, insects, do they have vertebral column, my dear children? No. But birds, mammals, fishes, they all have vertebral column, they are vertebrates. So, over here, the most species-rich taxonomic group among invertebrates is insects. Okay? So, we have so many types of insects. Now, the species area relationship was given by whom? Edward Wilson, Robert May, Alexander von Humboldt or Paul Ehrlich? Well, it is Alexander von Humboldt who gave the species richness relationship. So this is correct. Again, this chapter has more of the things which you have to remember rather than understand. You get it? Now, in the given species richness species area equation, log S is equals to log C plus Z log A. The letter Z represents what? The letter Z represents something that is known as regression coefficient. Okay? A represents area, C represents y-intercept. Okay, and S represents species richness. So it is a regression coefficient that is being represented by Z, my dear children. So I'll say this is A, this is C, and this is S in the equation. Okay, and this equation was put forward by Alexander von Humboldt. Now, the Rivet-Popper hypothesis was proposed by, which is a very famous and a very fun thing. So, Rivet-Popper, as in, he said that it was put down by Paul Ehrlich, okay, first thing. Now, he said that you can think of aeroplane as an ecosystem. Now, aeroplane has so many rivets, right? If you pop down each rivets, and he denoted that each rivet is basically a species. So, if you're popping down each rivets, what is happening? You are maintaining the balance of the plane, that is the ecosystem. Now, also, there are certain very important species. For example, the rivets on the wings. If you're removing that, obviously it will lead to a terrible crash. You get it? So, these species are also key species which are there, and you have to make them be there. Otherwise, the whole ecosystem would not be stable at all. So, Rivet-Popper hypothesis was proposed by Paul Ehrlich. My dear children, please remember it. Going ahead. Which of the following species introduced for aquaculture purposes is posing serious threats to indigenous catfishes in our country? So, the answer for this is Clarius garipenus. This was the one which was introduced, and it actually caused a lot of trouble for the indigenous catfishes. That means our own catfishes, our country's own catfishes. Okay, so this was an invasive thing. We got it from outside, and that was actually acting as a serious threat. Okay, this is not Ecornia, it is Ecornia, and then Parthenium and Lentana. Those are all plants. Okay, going ahead to the next question. The narrowly utilitarian arguments for conserving biodiversity are, that means there were different reasons why people were conserving biodiversity, and one such argument was basically we can conserve it because it provides us with certain products. You get it? For example, over here we have to look for the products. Either it can be in the form of medicine, or it can be form of food, or whatever thing is there. Okay, so cereals and fruits, obviously that is a product that we get from the nature, and since we are getting this product, we can obviously conserve the nature as well. Okay, now there are other things also, such as ecosystem services, intrinsic values, aesthetic pleasures, and things like that, but this becomes your answer. Okay, now 8th question. The following are the in situ conservation approaches except. So, in situ as in you are preserving them in their own habitat. So, there is an exception mentioned over here. We have to look for the exception. So, national parks, wildlife sanctuaries, biosphere reserves, all of them, basically in them we are preserving the organisms in their own habitat. But botanical garden is not like that. Over there we are taking the plants from their origin and putting it elsewhere so that we can preserve it. Okay, so this is an ex situ conservation, not an in situ conservation. So this is an exception, my dear little children. Next, Chanda and Bastar areas known for sacred groves are found in Meghalaya, Madhya Pradesh, Karnataka or Rajasthan. So Chanda and Bastar, my dear children, are sacred groves. What do you mean by sacred groves? As in, I have some religious attachment to a place, which is why I am preserving it. Now when I am preserving it, obviously that particular organism, whatever organisms and plants are there, they are also getting preserved. That is an example of sacred groves, and this is an example of in situ conservation as well. Okay, now over here Chanda and Bastar belongs to Madhya Pradesh. So please remember all the examples of different sacred groves. There are ones in Rajasthan, Karnataka, Meghalaya and all those things. So you should be remembering it. It's a very famous question that comes. Okay. Now question number 10. The World Summit on Sustainable Development was held in 2002 in where? Canada, Johannesburg, Brazil or Tokyo. So the World Summit was held in Johannesburg, my dear children, and it is a part of South Africa. Okay. So in that way also the question can be asked. So you should be knowing it is South Africa. And with this, we are completing all the 1 marker questions. Kudos to you all for being throughout and listening to all the questions, because obviously it is very much important for you to score really good marks. So I think it was beneficial for you. If you also think the same, definitely put down in the comment section, and lots of love, my dear children. You are going to do amazing in your exams. So bye bye. Take care.